Greetings to everyone, and thank you for joining our workshop today. My name is Jennifer Lippincott-Schwartz, and I'm a senior group leader here at Janelia. It's my pleasure to welcome you to our workshop focused on the cell biological basis of epithelial physiology. This is the second in a series of workshops that will help Janelia define a new research area focused on 4D cellular physiology, an area that will complement and expand our existing research programs on mechanistic cognitive neuroscience, molecular tools and imaging, and computation and theory. The workshops are being organized by me, along with Ron Vale, our executive director at Janelia, Nelson Spruceton, a senior director for scientific programs, Luke Lavis, senior group leader, and Janine Stevens, director of scientific programs. Many others have also provided helpful comments and contributions. 4D cellular physiology, or 4DCP for short, is the study of the structure and function of different cell types and tissues and addresses how cells interact to establish organ function and multi-organ coordination for animal survival and reproduction. It's a well-established field, but it seems to us that we are at a point in time when major advances in this field could be accelerated by taking advantage of the unique research environment at Janelia and developing new research approaches, data sets that will enable researchers around the globe. We feel that Janelia has an opportunity to contribute to 4DCP by bringing together people with varied expertise and ideas for collaborations in a very focused, highly supportive environment. We envision uniting for this endeavor not only biologists, but physicists, chemists, engineers, computational scientists, theorists, as well as tool developers and tool users with a range of backgrounds. We'll establish a 15-lab, 15-year effort in this area, which is significant but modest when put into a global perspective. Therefore, in order to maximize our impact on the field, we've decided to hold several workshops like the one today, which is focused on epithelial function, so we can learn about the state of the art in this area, as well as the conceptual and practical challenges that researchers face. This will help position us to focus our efforts on aspects of 4DCP where we can have the most impact. I'm very pleased to have all of you here today to help in this effort. The workshop was organized by me, together with Michael Caplan, who's a professor and chair of the Cellular and Physiology Department at Yale University, Patrick Chitwood, a postdoc in Ron Vale's lab here at Janelia, and Janine Stevens. We are fortunate to have a number of experts in the area of epithelial cell biology and physiology contributing their time, knowledge, and vision. We'll hear from them, as well as many early career scientists. The purpose of the workshop is different from that of a typical scientific conference. We want to answer a particular question. How can Janelia define its research mission in a way that will attract a talented cohort of scientists ready to tackle the challenges of accelerating different aspects of 4D cellular physiology? The workshop will consist of 4 sessions each day. Each session will have a few 10-minute talks followed by a 30-minute discussion. Now, unlike a conventional conference, these discussions will not just be Q&A. Instead, the discussion leaders will engage in a conversation amongst themselves, the other panelists, and with you. Please use the Q&A box to submit comments or questions that you'd like us to address in the discussion. Although we probably won't get to all of them, you can upvote the ones you like the most, and we'll use these to inform the ongoing dialogue that plays out during the discussion period. And be ready, because we might just invite you on screen to join the conversation. You can also use the chat box for more casual conversation and sharing of ideas during the workshop. I now want to turn the floor over to my co-organizer, Michael Caplan, who will provide some basics in epithelial physiology relevant to our discussions today. Michael.
So, hello everybody. I want to echo Jennifer's welcome and thank you all for joining us. And just begin by sharing a little bit about the biology of epithelial cells and talk to you about some thoughts that motivated this meeting. So, we all as biologists, as physiologists, are motivated by the dictum in some form or another that form follows function. And actually, that formulation was developed first not by a biologist, but by an architect, Louis Sullivan, who is known as the progenitor or the designer of the first modern skyscraper. In 1896, he published the following quote: "It is the pervading law of all things, organic and inorganic, that form forever follows function. This is the law." And it's interesting to note that that was published in Lippincott's magazine. I don't know if that's any relation to Jennifer or Presage's today's meeting. But what you see on the left is a columnar epithelium from gallbladder. On the right, basaltic columns from Giant's Causeway in Ireland. And what you can see is that forces at the macroscopic scale very much recapitulate at the microscopic scale that epithelial cells that form boundaries between the inside of us and outside of us very clearly recapitulate naturomorphic forms. So what are epithelial cells? As I mentioned, epithelial cells are the barriers between the internal and external compartments. We essentially are a complete tessellation of epithelial cells that control our insides and our outsides. All of the interfaces between the outside world and the inside of us are covered by epithelial cells. Perhaps most importantly, epithelial cells control the volume and composition of all of our body fluid compartments. It is not an exaggeration to say that what we are made of is determined by, defined by, and protected by our epithelial cells. Biologically, one of the things that makes epithelial cells most interesting is the fact that, like neurons, they are polarized. In the case of neurons, neuronal polarity is required for vectorial transport of information. In the case of epithelia, epithelial polarity is required for the vectorial transport of fluid, electrolytes, nutrients, often against extremely steep concentration gradients. The polarity in epithelial cells from a cell biologic or biochemical point of view can be seen when you look at the plasma membrane of epithelial cells, and you can immediately detect when you look at individual proteins that their surfaces are divided, their plasma membranes are divided into two distinct domains: an apical surface, which normally confronts a compartment that is contiguous with the outside world. So for example, the lumen of the gut, the lumen of the airway, lumens of kidney tubules, and a basolateral domain that rests on a basement membrane and is in communication with the extracellular fluid compartment. As you can see here, these apical and basolateral domains have very different protein compositions. And in order to develop and maintain this structure, epithelial cells need to be connected to one another by a variety of intercellular junctional complexes. There are tight junctions at the apical surface of most epithelia that form a barrier that prevents the outside world from mixing with the inside world. This is a selective transepithelial permeability barrier that allows some substances through, but not others. These tight junctions are also required to maintain the integrity of these polarized domains. A number of other junctions, some of which we'll hear about during the course of the meeting, glue epithelial cells to one another and are required for them to maintain their polarity. If you look carefully at these epithelial tight junctions here in freeze fracture, you can see this extraordinarily intricate array of particles that essentially form selective ion channels that permit, again, the transport of some substances and not the others, and again, recapitulate at a very microscopic scale the elegance of the structures of epithelial cells and the dictum that form follows function. The plasma membranes of polarized epithelial cells are endowed with a variety of morphological specializations that both reflect and mediate their functions. Some of them are shown here: apical microvilli, sensory cilia, motile cilia, which we'll hear about, I believe, from Lily Jan in the next talk, basolateral infoldings, among many others. Over the next 2 days, we're going to undertake an exploration of the big open questions that are related to epithelial cells. We're going to ask, what do they do? How do they form? How do they sense, communicate, and respond? How do their structures reflect their functions? How do their functions define their structures? And perhaps most importantly, for the sake of this meeting, how can we develop new tools and methods that will allow us to study them in their natural habitats? I'm very much looking forward to hearing everyone's contributions and to doing our best to think creatively to answer these questions. Thanks very much, and let's get the meeting started.
Okay. Okay, with that, let's get to the fun stuff. So the first session is titled Epithelia, Barriers that Beat, Clean, Sense, and Secrete. And we've got 3 speakers. The first will be Lily Jan, who's a professor at University of California, San Francisco. The second will be Gunnar Hansson, who's a professor at University of Gothenburg. And then the third speaker will be a postdoc from University of Gothenburg in the Pelasayed lab, and that is Elena Layunta Hernández. And then we will have a discussion following that led by Michael Caplan and Juanita from University of Arizona. And so let's turn over.
Thanks. For epithelia that we know is exposed to the environment, including the food we eat and the air we breathe, they form barriers to protect against pathogens, and they can also remove pathogens and via mucociliary transport or kill them. As a curious bystander of this field, I have questions rather than answers about how these epithelial cells form barriers and how the dynamics of microdomains and cilia movement contribute to barrier functions. As recent studies of epithelial ion channels have uncovered some rather unexpected functions, these surprising findings raise more questions. Some of those might be relevant to pathogenesis of human disease. So take the airway as an example. The epithelia, as Michael mentioned, contain both ciliated cells and secretory cells. And to protect against pathogens that may be in the lumen, exposed to the environment, secretory cells such as the goblet cells in the submucosal gland may secrete mucin for the removal of pathogens. And we will hear more about this from Gunnar's talk in this session. To replace or replenish epithelial cells, basal cells at the bottom, underneath the cilia, can produce both ciliated cells and secretory cells. A recent single-cell RNA-seq study led by Steve Quake and Mark Krasnov have identified 58 cell types in the human lung. These cells are marked by numbers in the schematics above, with the sections of the proximal and more distal part of the lung shown below. The same study also identified 41 cell types in the mouse lung that is much smaller. As summarized in this review by Michael Welsh and colleagues, bacteria in the airway lumen may be killed by antimicrobials, and they may be removed by mucociliary transport. CFTR chloride channel that is linked to cystic fibrosis permeate not just chloride ions, but also bicarbonate that may affect the pH of air surface liquid. And besides CFTR, there are calcium activated chloride channels also on the apical surface, as shown by electrophysiological studies since the 1990s. These observations raise the prospect that increasing calcium activated chloride channel activity might remedy some of the problems resulting from a reduction in CFTR chloride channel activity in cystic fibrosis. To pursue this possibility, the Enterprise Therapeutics in UK recently developed potentiators of TMEM16A calcium activated chloride channel. These potentiators not only increase the calcium activated chloride current, but also the height of airway surface liquid secreted by human bronchial epithelia from cystic fibrosis patients. We can see this effect 1 hour after exposure to the potentiator, and still a greater effect after 3 hours of stress that's simulating tidal breathing. With these promising effects, both in vitro and in vivo, the TMEM16A potentiators have recently been acquired by the Roche to explore their potential use in treatment of cystic fibrosis. Besides problems due to a reduction of chloride channel activity in the airway, infants with cystic fibrosis may have congenital airway defects. This tracheomalacia or something very similar to what's seen in the infants is also seen in the TMEM16A knockout mice, as reported by Jason Rock and Brian Harvey. So for some reason, these chloride channels are important for airway development. Mu He, who did her postdoc research in our group at UCSF, has found that TMEM16A is also important for proper cilia movement in the mouse trachea. The fluorescent beads placed on the luminal side of a sheet of wild-type mouse trachea will move in the distal to proximal direction, while these beads will move in all kinds of directions when placed on the luminal side of TMEM16A knockout mouse trachea. This probably reflects abnormalities in the epithelial cell organization and cilia movement. In collaboration with Michael Caplan and Spiros Darmantis at the BioHub, we could see a multitude of knockout mutant phenotypes, including mucus obstruction, simplified alveoli, excess secretory cells, shortened microvilli and cilia, dilated ER, accumulation of large vesicles, abnormal expression of immune modulators, as well as defective adherence junction remodeling. During embryonic development, the airway epithelia progress from predominantly pentagons at E14.5 to predominantly hexagons at E18.5. This process involves endocytosis and recycling of adherence junction proteins, and it somehow depends on TMEM16A. Rearrangement of epithelia also happens in adult tissues, and for airway epithelia to function as a barrier, it will be interesting to see how it maintains epithelial integrity while incorporating cells produced by the basal stem cells. For the polarized epithelial cells, they rely on membrane traffic regulation for the adjustment of adherence junctions. They also rely on membrane traffic for the polarized distribution of transport proteins. These ion channels and other transporters can facilitate the movement of water and small molecules as well as ions to regulate secretion and cell shape. And they provide yet other functions. For example, chloride channels have been implicated in the transport transepithelially of thiocyanate, which is used to generate the antimicrobial hypocyanate. While we don't have the biosynthetic capacity to make SCN, we can get it from food like broccoli. And TMEM16A chloride function is also important for the control of membrane composition, as in the case of phosphoinositides. And with a change in chloride concentration, we could see rapid rearrangement of microdomains of PIP2. While these observations help to connect chloride homeostasis with cell biological functions, such as trafficking of recycling endosomes, we still don't know the underlying molecular basis. So where to go from here? It would be nice to be able to follow the dynamics of epithelial cellular components, the movement of these cells and their reorganizations in more physiological setting. What could go wrong? Besides compromised barrier functions, as in the case of cystic fibrosis, other problems could arise from infection of epithelial cells. The autopsies from last year from COVID-19 reveal the presence of syncytia. So these are cell fusion events that involve lung cells that express a surfactant and also the transcription factor TTF1, and they involve infected cells as evident from the presence of SARS-CoV-2 RNA and protein. While fusion of infected cells with surrounding cells may provide a way for virus infection and replication to persist for a long time, it poses yet more questions about epithelial cell pathogenesis. Thank you.
Okay, it's my turn, Gunnar Hansson from Sweden. I will talk about mucus and mucins and really what is outside of the epithelia, but of course made by the epithelia. Mucus is kind of very unexplored areas, and I would like to try to give you some concepts of how I think this works. And I will then use our studies of the intestine and the lungs to do that. And mucus has really two functions. It is to coat surfaces of the body, and in doing that in two really ways. One way is to clean and wash away things, to not let them attack us. And the second way is to coat physical surfaces to generate a protective system. And in fact, it is like this, that in the small intestine in the normal way and in the normal lung, you really trying to move things, whereas in the stomach and the colon and in the lungs under disease conditions, you will instead do a protective system. And I will try to show a little bit more about that now. So we start with the respiratory tract in the normal way. Then you have submucosal glands. You're making mucins, which are secreted out from the goblet cells. And these are then passing out and forming thick bundles, which comes out on the surface, where you see the cilia, which is beating, as you see on the next picture. These mucins are packed in a very, very specific way, in such a way that when you pull them out, they form long threads, which is then coming out of the glands, as you can see here. And mucus is a lot of water, and that is why whatever you do to fix them, they will usually shrink, and you don't see them as they are in native state. If you then go to the live tissue, you can see here how these bundles, when they come out on the surface, they are sweeping the surface. But the interesting thing is that the flow is in this direction up, but still they're moving in a perpendicular direction, which we don't understand a thing about. And in this animation here, you can see how these bundles are sweeping. You see the cilia in the background, you see the liquid floating on top, and you see how they sweep and take the microbiota with them and clean the surface behind it. So in the normal lungs, there isn't really very much of a mucus coating. And there is also thinner types of mucins coming out or like threads out from the goblet cells. So there's a number of questions here which we have no clue how that works and how that functions. We can see that when we stain coming out of the glands, you have this MUC5B mucin, which I showed, but that becomes quickly coated by another type of mucin which is called 5AC. And the interesting thing is that in the case of cystic fibrosis, these bundles don't move at all. In fact, from the newborn kids, they are still staying, and you see much more attachment site. So there is something here that you're holding down this type of bundles onto the surface. How this works, we have no clue at all. Different thing is when you look in the mouse, then it is also washing away, but then mouse is smaller and you're not making these bundles because you don't have any glands. Instead, you're making clouds of mucus, which is transported again up against the gravity towards the larynx. So again, cleaning and washing away things. If we then go to the small intestine, where you also have a cleaning system, in the normal way, you see now pointing against you is the villi, and which put charcoal on top of it. And in the normal case, you are able to aspirate away the mucus, and with that the charcoal, which is on the top. And this is how it should work in the normal way. It is that the mucus should be formed and it's coating the whole surface, and then it is washed away by the peristaltics. But if you then, like in the cystic fibrosis channel, CFTR channel in the cystic fibrosis, you see here that the mucus is not freely movable. It is still anchored. It is anchored to the epithelia, and you can poke it around, which means that this movement of mucus is controlled from the host in some ways. And the CFTR channel is a master regulator of these type of things. If we then come to the other aspect of mucus is to protect. And this is what we discovered some 10 years ago or so, is that there is in the colon, there is a mucus layer which is attached to the epithelial cells. And this is around 50 micrometers in the mouse and 250 micrometers in the humans, which is more or less impermeable to bacteria. So you keep the bacteria at a distance. So this is a protective system. And by that, you can keep the bacteria away, and they don't come in a normal contact. If you have a system or something happened to this protective system, so it is not that impermeable, which it is in for example, when you get ulcerative colitis disease, then this system kind of partly falls apart, and bacteria can penetrate down to the epithelia. And there's a lot of questions we don't understand here. How is the mucus attached, control the mucus release, and how is it organized? And you come to the lungs. I told you before that in the normal lungs, you don't have any mucus coating, but instead you have. But if you get bacteria on top like this, then you get an expansion of the number of goblet cells, transformation into goblet cells, and then you're making a mucus layer, which is in fact attached in the same way as it is in the colon. And in the normal case, by that you are moving bacteria away from you. But if you are healthy, then you are turning on the CFTR channel and you are coughing it up and you're cleaning the system. But if you are not healthy, if you are if you stay like this, like you do in cystic fibrosis or in the COPD, you generate a mucus layer which is coating the surface the whole time, and bacteria will stay here. And of course, by time, even if the lungs are better protected than normally, the bacteria will slowly generate inflammation and the lungs are destroyed. So I think this is a kind of a general type of reacting for the lungs. And if you look on the respiratory tract, you can see that in the cystic fibrosis, both in pigs and in humans, you can see, this is from Mike Wells' lab, and you can see here that you have a stratified system. You have the same thing in the COPD and also in experimental system. And what it is that you have the mucus attached to the goblet cells and it stays and it sits attached to the surface. An interesting thing is if you have a mouse and you introduce bacteria, in the normal case, the bacteria can come in direct contact with the epithelia. These are the nuclei with the blue here. They come in direct contact. But if you first induce a mucus layer in the colon, in the lungs, you can see that the bacteria is trapped in the mucus and they don't really reach down to the epithelia. So you generated a protective system. So the key to all of this is the goblet cells. The goblet cells are the ones that are producing the mucus. And these are very specialized cells. And the interesting thing with them is that they're very unexplored and considered to be very dull and non-interesting cells. But it turns out, as you will see, that there is a number of different goblet cells with very specialized functions. And in fact, there is a few things which we know about now. They know they have a special chaperone systems that can make mucus, but we don't fully understand how that works. There is an interesting system controlling the length of the mucus, which is totally not understandable because it means more or less measuring several thousand of amino acids, how long they are. And it is also translational control. There's also very dynamic glycosylation machinery, and as you will see shortly, the granule here and the secretory mechanism is totally different from what we know so far. And this is showing, I would just like to mention also that what you do in the goblet cells, you send them out to free space. What you have packed with your mucus when you send it out from the cells is what you have. You can't do anything. Most of cell biology goes in cells, then you can have control system and so on. Between cells, you can also, inside the body, you can have control system. Whereas when it comes to mucus, you have to send everything with them when they come out. You're sending out something to the space shuttle, space stations out there. If you have forgot something, it is too late to do anything. And this is now from a gland, from a crypt in the small intestine. And here you can see when you stimulate by carbachol how the mucins are released from the goblet cells and how they are washed out. And in fact, it turns out that this is a collaboration between the goblet cells and the adjacent cells. You need to communicate between them, because if you block the gap junctions, you will empty you see the stimulation in the goblet cells, but you won't clean as you see. And if you're lacking the CFTR channel, you also see that you get the granule in the goblet cells are starting to become released, but you don't wash out. So you need to have a combination of different cells to make a proper mucus coating of the body. We have now recently done single cell analysis of, this is from colon. Malin Johansson has been heading this, and it turns out that there is a number of different type of goblet cells with different setups there is a canonical system, and there is a more enterocyte type, and with some very specialized cells which is up on the surface. And finally, I would just like to show you some recent exciting data from Brendan Dolan in the lab. It is that looking at secretion using stained Mark II mucin, you can see that the granule disappear here, and they are releasing and exploding. But it turned out, when you look into detail, what is happening is that the first thing after stimulation is that the granule starts to expand in volume, so they change in their nature. And what happens then is that the granule explodes, or inside the cell, you still have an intact membrane which is coating the surface, and finally the whole cell explodes. So you get a rupture of the luminal membrane. This is a totally different secretion than we have seen before. So goblet cells are very specialized cells that can do very special things. So by that, thank you very much. And here is part of the people. So thank you.
Okay, and with that, we'll move to our next talk from Elena.
Hello, everybody. Thank you very much first for inviting me to give a talk here. I am Elena Layunta. I am at the Pelasayed lab at the University of Gothenburg. And we are going to talk today about one of our main projects, that is the role of intestinal glycocalyx in Crohn's disease. Yeah, so why we are studying Crohn's disease? The Crohn's disease is an inflammatory bowel disease that we don't know the cause, and we don't know the etiology, and there's no cure. It's including in a chronic inflammation that affects mainly the last part of the small intestine, and there's a significant increase in the incidence. Most of these patients from Crohn's disease, they are infected with adherent and intracellular bacteria. So we don't know, and there's an inability of the intestinal epithelial cells to resist and eliminate these intracellular bacteria. And we have this big question, how the intestinal epithelial cells can protect the host against these bacteria in the intestinal lumen, and is what we are working in our lab, how the enterocytes can protect the intestinal epithelium. So as Gunnar says before, in the small intestine, we have a mucus layer that, in a difference with the colon, is permeable to the bacteria. And these bacteria can reach the intestinal epithelium, where they find, the first thing that they find is the glycocalyx. The glycocalyx is a group of glycoproteins and glycolipids that protect us from this bacteria in the lumen, and where the main glycoprotein is the membrane mucin MAC17 that we think that sends this intracellular, this bacteria in the lumen and trigger defense responses, intracellular responses. Some highlights from this glycocalyx is that it's established during the weaning, that is developed by innate immune signals, and also that prevents bacterial adhesion. And you can find more in our paper that it has been recently published in Cell Reports. So we know that in the Crohn's disease patients, there's a problem in the adhesion of this bacteria and trigger different inflammation in the last part of the ileum. And we have really seen that this glycocalyx protects us from this bacteria. So we thought that maybe there's some defects in the glycocalyx in these Crohn's disease patients, and it's our main question, or big question in this project. So we saw that the main membrane mucin of the glycocalyx, MAC17, that you can see here in green, is well expressed in non-IBD patients, but in Crohn's disease patients, there's no MAC17. So it seems that there's defect in the MAC17 and therefore maybe in the glycocalyx. However, with these classical methods of fixation, as Gunnar says, we dehydrate the glycocalyx and we are not able to study the function of this important barrier. So we developed a new method that is called glycocalyx permeability assay. Briefly, we take live biopsies from the intestine of the patients, and we incubate with different label molecules, for example, fluorescent dextrans, or also label bacteria as positive GFP E. coli. As you can see here in the pictures, we can see in green the 2000 kilodalton dextran and also the GFP positive E. coli. And in gray, we can see the cell mask that it stains the limits of the intestinal epithelial cells. So what we analyze is the colocalization of these molecules, of these fluorescent molecules, with the cell mask, with the limits of the intestinal epithelium. And then we assess the permeability of the glycocalyx. So we saw that in Crohn's disease patients, as we hypothesized, there's more permeability of the glycocalyx, as you can see here in the dextran studies, and also more encountering of these bacteria in this assays. This method that we have developed in our lab give us great opportunities. First, we can define the impact of molecular perturbations at enterocytic surface on cellular barrier function, what it means. So we can assess the treatment response of these patients in Crohn's disease, pathology. Moreover, we can study the glycocalyx barrier in different knockout mice models, as for example, the ones that we generated here in the Pelasayed lab, the MAX17 conditional knockout, also other models of abnormal oglycosylation because the glycocalyx is high glycosylated, so defects in the glycocalyx by these knockouts can generate different studies further. And then the orthogonal glycan leveling of the glycocalyx as the injection with GalNAs. Moreover, and apart from the Crohn's disease, we can study other pathologies in the intestinal tract, as the ulcerative colitis, but also irritable bowel syndrome, and also intestinal infections by bacteria. Because this approach is really novel, we have really several limitations or challenges. First of all, as Gunnar says, there's mucus in the small intestine that, of course, they protect us against the bacteria. So sometimes when we take the biopsies and then we try to get rid out of this mucus to study the glycocalyx, that barrier, we try not to affect the architecture of the intestinal epithelium, which is really tricky. Other important limitation is that the preservation of the tissue is really tedious, and we have to really do it really fast, and also to preserve the native properties of the membrane mucins are really difficult. And finally, we are working in the development of softwares and plugins to analyze in an automatical way this glycocalyx permeability assay to be it in an accurate way and also in an automatically form. So I want to thank everybody in the lab, in the mucin biology groups, especially in the Pelasayed lab. So first, my boss, Tahir, and also my colleagues, Anandi, Sofia, and Gustav. And of course, all the Crohn's disease patients and the clinicians at the Sahlgrenska Hospital at Gothenburg, because they lend us all the material to study and to carry out this novel method that we have developed. Thank you very much for your attention, and yeah, we can discuss now and then resolve all the questions that come up. Thanks.
Great, thank you. I'd just like to thank all of the speakers, and then now I think we can turn it over to Michael and Juanita for the discussion.
Yes, thank you. Those were great presentations. There's actually a couple of Q&As from the audience, and I would say the first one from Paul Welling that I actually would like to dovetail on myself. So the question is, do the different goblet cells defined by scRNA have different functions. Do they represent one cell type at different stages of development or physiologic control?
I mean, to be really honest, we don't have the fully clear-cut answer to that, but it's clear that we have two trajectories at least. So there are at least two major lines of goblet cells. One is, which is more a typical canonical type of goblet cells, which are more what you consider as people should think is a goblet cell. Then there is one which is much more enterocyte-like, but both all of them change, and we don't know really how if they change during they dividing in the bottom of the crypt and then they migrate up, but exactly how they if they can differentiate from one to the other, we really don't know. But there are a number of very specialized goblet cells. We published in Science a couple of years ago, about the sentinel goblet cell, which is one goblet cell sitting in the crypt opening of the colon, which is sensing if the bacteria comes closer, and then it signals to the adjacent goblet cells to secrete. And we have a new paper coming out in Science shortly talking about the intercrypt goblet cells, which are sitting between the crypt and the colon, which are totally different because they are secreting continuously and they are don't accumulate. So in fact, they don't have very big granules, and that means that some people wouldn't call them goblet cells. But we have decided to define a goblet cells which is producing mucins. And since we've made a couple of different transgenic animals with different colors of the mucins, we can easily isolate these and study how they are. But I should say that there are at least two trajectories, and there is they differentiate. But if every cell can do different to be one, for example, intercrypt goblet cell, we don't really understand exactly how that works. We're not there yet.
Can I just jump in and just dovetail on this whole idea of the different types of goblet cells? What actually determines the number of goblet cells per gland? And I think this is particularly important in the lung, where you probably don't want to have so many goblet cells that alveoli are going to fill up with mucus. Perhaps in the small intestine or in the intestine, it's going to be not such a critical issue.
I mean, it is really, a lot of it is determined by cytokines. For example, IL-13 is a very important one for determining the number of goblet cells in the lungs, because normally in a very healthy mouse, in a very dust-free environment, you really see very few goblet cells. And almost none. Whereas as soon as you stimulate with dust and things like that, then they start to come up. So it is, overall, I should call the goblet cells a totally ignored cell. Very few people has looked at it. And what they're doing most of the time is looking for cells filled with this granular. But that is just part of it. For example, if you have the disease ulcerative colitis, then they say you have goblet cells depletion, is what people say. But that's not true. The goblet cells are still there, but they're emptied.
Okay, what about the Notch signaling, I think, and also controls?
Yeah. But that is determining the Notch and the Wnt signaling is of course studied by, and that is determining which lineage you're going and of course determining which way you're going. That's right, of course.
Okay. All right. Mike, did you want to, we have a hand up, and we have quite a list of questions developing.
Yeah, so maybe the next question we can move on to is one that I also had, is do goblet cells self-destruct when they expand? In other words, are goblet cells, does secretion essentially kill a goblet cell? Does it need to be replaced, or can it be repaired?
I mean, for sure, some goblet cells are not killed. But for example, the sentinel goblet cells, which we published on, is once it's been secreted, then it's pushed out and out into the lumen. So that one is killed by a cell, whereas others are not. So I should say it varies. Some can do it, some cannot do it. The thing which I showed with this kind of secretory mechanism is, I don't think that necessarily kill them, because you can find goblet cells which are almost, look like they're dead and with a halo more or less in them, but they still can survive. So there are mechanisms of repairing and retrieving a lot of the membrane, and so goblet cells are also very much endocytosing, which you might have heard about, that there are so-called goblet cells in the small intestine which are retrieving material from the lumen and delivering to the dendritic cell on the other side to generate tolerance. So there are mechanisms of retrieving a lot of the membrane which you lost when you're doing that, and I don't think you're killing the cells, but of course, for sure, we don't really know. We haven't studied that in detail.
Thanks. Juanita, you want to take
Yeah, so I actually, Mark von Zostro, I'm sorry, I know we have a hand up too, but just to follow up on basically the RNA-seq phenotyping of these goblet cells. So he's asking the question, can you say more about the RNA-seq analysis and its depth? Specifically, I wonder if you have been able to assess the combinations of G protein coupled receptors and receptor tyrosine kinases co-expressed in the cell types resolved.
That was a too specific question to answer out of, I can't really, I mean, we have done both single cell and RNA-seq. So with RNA-seq, we have quite deep, but when it comes to the other one, we are down to 4,000 transcripts. So it's not complete, of course, but that specific question I can't answer.
Okay, but sort of along the lines of what you said in terms of determining the number of goblet cells, you had mentioned that this is controlled by cytokines. So I'm assuming that it's going to be several of these types of cytokine pathways. Are these receptors on the apical surface or primarily on the basolateral surface? So the polarity also may play into. You probably can't tell that from the bulk RNA-seq though.
No, but I should say that it depends a little bit on I think that that which of course one of the issues is how much is signaling from the outside, and the goblet cells in the intestine is very much dependent on the bacteria. Mm-hmm. So and if that is direct or indirect, we really don't know very much about that. When it comes to the lungs, you have a luminal macrophage, for example, patrolling the surface there. So there you have another system which can signal from that side. Either, of course, a lot of signaling come from the basolateral side, of course, yes.
Okay, let's go to Prav Sengupta, who's had his hand up for a while.
Hi, great talks. I have a general question about where we are in terms of understanding the form and function of these intricate and complex epithelial tissues, which line the airways and the lungs and the intestine. Like, for example, I guess to really have a comprehensive understanding of how these tissues work, one of the criteria is that we need to identify all the players, right? Now, Lily showed that there's a very diverse array of cells in the epithelium layer of airways, and like ciliated cells, goblet cells, and I'm guessing since this epithelium layer plays a central role as the first line of defense against bacteria and organisms, the cells must be interfacing with immune cells and probably also nerve cells. So, and probably this is true for the epithelium lining the gut and other epithelium as well. Now, the question is that I had for the panelists is that, like, in order to understand how all these diverse cells are organized in 3D in these tissues, like we would need a high resolution mapping, like a morphological mapping, probably with some kind of an electron microscopy based method, but that probably would not be enough, right? Because these cells probably have different cell states. So we also probably will need some kind of a multiplexed single cell analysis to get a map out the transcriptome, the protein expression, perhaps even lipidomics, whilst being able to preserve the spatial positioning of these cells in the tissues, right? So the question is, that's one way where actually, I guess, a place like Janelia can invest in to create that map, that very high resolution functional map. Now, where are we basically in, like, is that something that basically is worth doing? And then the other question is that in order to then go to the next level of understanding, what probably we need to know is basically how these cells are actually communicating and interacting, and how that's actually driving the basically new and emergent properties at the community level, right? So, and for that, we also probably need sensors, which for this particular cell type. Now, my basically general question is that we can do both, or we can probably basically concentrate on one of them if the first part is already in a more advanced stage. So so I would like the panelists to basically maybe comment on where we are in terms of understanding these questions.
So Lily, you had your hand up. Why don't you respond to Prab's?
Yeah, so good point. So one thing that would be really nice is to bring the single cell RNA-seq to the level where you can see the different cells, in tissue, rather than after dissociation. There are efforts for doing that. It may be still early stages, and it will be nice for different ideas to come together or separate in parallel to make that dream happen. And the other question about communication, I was too ambitious. In the pre-meeting, Michael said, "Oh, you prepared 2 talks, take away 1." And so, because one thing that I thought would be a real challenge that the Janelia farm, approach may really help is the question about communication. There are different possibilities, secreted molecules, extracellular vesicles. And for polarized epithelia, the vesicles secreted from one side or the other may go to different cell types. And those are really tiny, so it's a real challenge to see them in physiological settings.
Can we jump over to one of our panelists, Elaine Fuchs has a question. Elaine?
Yeah, this is really fascinating. Gunnar, as you were presenting your talk, I was thinking about Richard Loxley's work, where he showed that in the intestine, the helminths, the worms in the intestine, can trigger through changing their metabolites, and the metabolites in the milieu can affect these certain immune cells called ILC2s, and that then secrete IL-13, which you mentioned also, and that impacts on the intestinal stem cells, and they skew their fate to make more goblet cells and make more tough cells, more of the cells that are going to fight the the infection. And I'm curious to know, with regards to the mucin that you see, it seems like there's 2 different ways then. One is to up the level of goblet cells, and the other is to secrete more mucin. And in your case, where you had this bacteria secreting the mucin, it seemed like, the bacteria stimulating the mucin secretion. I wonder, do worm infections do the same thing, or are worms can they just go right through the mucin?
They are, I think all type of pathogens, all commensals, they have ways of communicating with us. They have developed together with us. And for example, if you take a germ-free mice, they have the same mucus system as I described, an inner mucus layer and an outer mucus layer of colon, but it's totally penetrable to bacteria. But then if you put in bacteria, then it starts to change its properties. And the interesting thing is that the composition, when it comes to proteomics simple, is not such a different thing. It is rather ion channels and other things which is happening during the formation of this, which you have no clue how that works. And it is, I think, also very important to remember that this is not the work of one cell. It is the work of the whole cell system. They have to communicate with each other and to do the proper work that they should do. And I should say that goblet cells have probably one of the biggest challenges to do this type of very complicated molecules, and that's probably why they have a number of chaperone systems, for example, which is very specific for that. We have mucins which we can't produce in any cells because we don't understand how it works. And of course, these type of molecules, these type of gel-forming mucins, they developed in the metazoan early when we started to get the most primitive intestine, and they have been with us all the time. And of course, they've been developed a lot of things together, and which we have no clue exactly how that works.
So following up on that, I'm sorry, Elaine, did you want to ask a follow-up?
I just wanted to follow up in just a very brief thing. Are there pathogens that eat up mucin?
Yes. But they are very, I mean, we have a new one now, which we found it is very specific, and it is very elaborate how it's made, because a lot of the thing, how you avoid being eaten up by pathogens, is that you're using the glycans, because you decorate and you change that. You can change that a lot, so you can. But then there is one, the ETEC, they have a very specific enzyme which is recognizing a folded piece of a mucin and cleaves and destroys. So it degrades the whole system. It do that in the human sequence, but it doesn't do it in the mouse sequence. So there's a lot of things built in here. Of course, we have to think about, we have been battling in over evolution here with different pathogens here and there, and that is part of the reason why some are pathogens. I mean, if you are a pathogen, you are able to destroy the system. If you are commensal, you don't do that, because then you live on it.
So thanks. It's clear that there is a real dialogue going on between epithelial cells and the things that are attacking them and the stresses they experience. And clearly the presence or absence of bacteria play a role in determining the number and differentiation of the mucus cells. As Lily showed, ion channels in the epithelial cells themselves can modulate the number of mucus epithelial cells that form. So the properties of the epithelials, the columnar epithelial cells, as well as the environment they find themselves in, plays a role in defining the quality and quantity of mucus. And as Elena showed, the glycocalyx plays a critical role in that as well. I'm wondering if we can bring Manu in to ask a question that's directly relevant to this, relating to how the cilia flow changes in the mutants that she talked about. So Manu, do you want to ask your question, or would you like me to ask it for you? All right, I'll go ahead and ask it for him. So Manu asks, the mutants that you showed with reversal of ciliary flows, is that a breakdown in orientation or in the number of ciliated cells? And I will add a corollary to that. How did the presence or absence of chloride channels in the epithelial cells play a role in changing either ciliary flow or differentiation, or both?
So I have a partial answer, really. What we can see is the length of all kinds of cilia, primary cilia, motile cilia, and also microvilli is much shorter in the mutants. And from the single cell analysis, it looks like there's increased number of secretory cells, but not increased number of ciliated cells. We were following the fluorescent beads. It would be really nice to see the cilia movement, to see which way they are beating in these tracheal explants, the way Gunnar has shown for the cilia movement. What happened? Why are the cilia shorter to begin with? Part of it may be the membrane traffic, the formation of cilia. And we have also seen, the tips of primary cilium just dropping off. That's why we got so drawn into the extracellular vesicles. They came off as extracellular vesicles.
Great. Juanita, do you want to pick out a couple of more questions from the
yes, I sort of like to, and I see Patrick has had his hand up, and also Jitu, but maybe we can talk about the goblet cells in more than just the intestine and the respiratory tract. There's a couple of questions that we're talking about that, and then also how these cells communicate not only with epithelial cells, but perhaps the immune system. So I'll throw that out there, but maybe Jitu, I think you had your hand up for a while. Did you want to ask a question? Maybe pick up those concepts?
Yeah, thanks, Juanita. Yeah, no, I have a question, in fact, for both the systems that we are listening to. I mean, they're of course incredibly complex, these with multiple cell types and many, many different types of interactions. I was just wondering, I mean, can one build this system up? Are there good organoid models? I mean, how does one account for I'm sure the incredibly important role that mechanics is going to play in the way the systems are built? I mean, can we think of ways of, so in some sense, engineering these types of tissues to create the complexity that you've so beautifully described? So it's a question for, in fact, both of the people who are working with both of these systems.
If I can answer first, I mean, yes, I mean, these endoid systems, I mean, which has been developed, I mean, they are really very good at mimicking the stem cell niche and how they move up along the crypt and that. But of course, it's not really there yet to say that you can mimic the whole system to form the surface cells, and I think it needs quite a lot of more work to get there, and really, for example, to generate endoids, to generate a 2D system where you get a layer of cells, so you can get a mucus surface on that. I mean, there are people claiming that they can do that, but it is not really, we are not there yet. And it also needs probably physical stimulation by movement and things like that. And of course, in the intestine, we have the peristaltics, which I think is extremely important as well. And of course, in the lungs, you need to have the flow of the cilia. So we are far from being, but I mean, of course, as all systems, you can always study certain parts in an artificial system. That's where we are. But to generate the whole system, we are not there yet, really.
Can I jump in and maybe call on Jim Wells, who actually works quite a bit on these organoids and generating these interoids from iPSCs. Jim, did you want to make any comment about using the sort of mini gut systems to study goblet cells?
Yeah. It's certainly something that people do use, different organoid systems. There was a beautiful recent paper, which I'll mention briefly tomorrow during my presentation, of different model systems, but basically allowing to look at dynamic remodeling of both the epithelia and probably the mucus layer as well in a bioengineered system out of Matthias Lutolf's lab in Switzerland. So that there are emerging model systems that should allow you to both study mucus production, response to various types of bacteria with luminal flow, so in a much more dynamic way. So yeah, these systems are coming online and should allow for really more detailed studies of goblet cells.
Important tools, yeah. So that's going to definitely get important.
This could be a great area to commit some energies in. I think the engineering of these systems.
So along those lines, perhaps, maybe, Zev, if you could ask your question, because it relates to the context in which the mucus secreting cells find themselves.
Yeah, thanks. Oh, wow, frog in my throat. Yeah, so my question, I was thinking about the sort of steady state dynamics of the epithelium and how cells move up the crypt-villus axis. And thinking about the flow of the mucins, do you think about the goblet cells as moving along with the rest of the cells as they undergo differentiation up to the tips, or are they more like islands in that sea of cells? That's my question.
I mean, what we know is that they are of course moving up, but they are not moving, together. Certain cells are moving faster than others, and some are staying. So they interact, and of course, what should be interesting to know is what stage they are determined, what kind of fate they will have in the future, what type of cells will. But with that, we are, maybe what we should need to have them is what was Lily pointed out, to have a single cell in situ, so we could know more what kind of programs they have, because for the moment we just see them as collectively one type of group of cells, and of course this is a very quick system. We have measured the renewal rate of in the mucus layer, which is it's 1 hour. So the whole system is extremely fast in renewing itself, and these glycocalyx with Elena talked about, that is a little bit longer, but that depends also where you are, if you are, how far up you are on the villi system, and so there is a number of things here which we also need to understand better to see how these systems are working and how they are protecting us.
So Elena, can you comment on how the glycocalyx changes as you go from the crypt to the villus?
Yeah, exactly. So we just recently saw that this development of the glycocalyx starts in the crypts of the villus, and then it comes up and then covers the whole villi. So it seems that during the infancy, there's a really opportunity for the microbiota, for the beneficial microbiota to create this glycocalyx, but it's created from the crypts of the villi. So since that all starts from there and then covers all the villi, so it's we don't know yet what's the different functions of the apical or the crypt. We wanted to go further in this with the glycocalyx, but it seems that there's a difference there.
Thanks. And Patrick and Jennifer, you've had your hands up for a while.
Yeah, I guess I have a general question for all the panelists, which is that it seems like a general theme that kind of emerged through the three talks was that in response to some physiological cue, you could have a very very specific adjustment in cell type to elicit some very specific response. And I'm just curious, how exactly do you adjust a cell so dramatically to then elicit this response, if that's well known, and what would be the best way to interrogate that? So like how you would actually change a cell in response to a cue, and now it can become, a massive mucin secreting cell, and also changing the sort of makeup of an epithelial in general to now contain more of one cell type versus another, and how that's all sort of put together and how you would actually interrogate that.
I don't know who you want to answer, but it's I think that the lung system is an interesting system to really address that, because there you, as I try to convince you about, is that it's got one system which is there in the normal case, and then you can generate a totally different, more protective type of system, and of course how the tissue responds to that and how it develops that. It is, we know, of course, that there are some cytokines we can do that, but exactly how that reacts to, does for example, the cilia, when they're beating, do they sense that there are something more difficult to move? There is a lot of things we don't understand. How this system could change, and which I think is an opportunity, of course, to try to address communication from outside in, which is probably one of the least understood things in biology.
Right. I guess a couple of things. One thing that was once mentioned in Lily's talk and also Gunnar's talk was the single cell sequencing to identify, specific cell types and identities, which I guess gives you a little bit of a handle on trying to figure out what's different about those cells. But then I guess in terms of tools, I guess what you'd really want to know is, or what you'd really want is to try and label a specific cell type so that you can study that specifically by imaging or even biochemically. And so, is it really that these cells are dramatically expressing different things that you could use as a handle, or is it just a minor adjustment of an already existing infrastructure, making it much more difficult to sort of interrogate that particular change?
As I mentioned, we have tagged 3 of the 4 gel-forming mucins, and Elena has tried to, but they didn't manage to make the Mark 17 label. But it's possible to do that in a certain way. But of course, that doesn't mean that you can separate out different type of goblet cells, for example. We get all goblet cells. And together, I will also say that one shouldn't look only on one cell, because it's extremely important, the collaboration with other neighboring cells. And I've been spending my whole life on mucins, but I realized that ion channels is probably maybe even more important in regulating the properties of these type of molecules. And so you need to have a really broad type of people to work on this with different competences and different angles to get into this type of epithelia.
Right, and we haven't spent a lot of time on the tool aspect. And one of the things I was going to throw out there, I think, in terms of labeling these cells and these molecules, there's typically the genetic approach, but no one has really talked about like a penocytosis type of approach where there may be particular goblet cell types that will actually take up some of the lumen, maybe labeling the bacteria so that you can follow specific goblet cell populations. Let's go to Jennifer for our last question, because we're pretty much at the end.
Well, I'm going to just punt over to Thomas, but the one thing that I would like people to think about, 2 weeks ago, we had a workshop on the brain body where everything we thought about was neurons and how neurons are communicating with different tissue systems. I would like to know how you guys think neurons are involved in what we've just discussed. But now I want Thomas to quickly throw out his comments. Yeah.
Oh, yeah, I will say we've just run out of time on the discussion end. So we have probably time for just one more comment by Thomas, and then we can move on to the next session.
Thanks.
Yeah, it's a very quick question. I'm curious about the growth potential of these organoids in vitro. How do you sustain growth for days, weeks? I mean, we know in vivo that they are at steady state, and so there's cellular renewal, but really, literally, there is constant growth. So how can you supply that in vitro?
Jim, did you want to take that?
Maybe table that question until after my presentation, if you don't mind, and then we can discuss it then.
Okay.
Yes, ask Jennifer's talk. Yeah, I mean, you know, the intestine is the most neuron-rich organ we have in the body, except for the brain. So it's
okay. We'll talk more about that.
Yeah.
Okay.
And it's really related with the brain, by the way.
Good.
Great. Okay. With that, I think we should move on to our next session, which will be introduced by Michael Caplan, and I'd just like to thank all the speakers and also thank the discussion leaders involved for a great session.
Thanks. So that was a terrific session. It set a high bar both for the talks and for the discussion. This second session is entitled Integrating Across Scales and features talks by Francis Ashcroft from University of Oxford, Allison Zajac from University of Chicago, Helen McNeill from Washington University, and Eichmann from Yale, and will be moderated by Mahadevan at Harvard and Manu Prakash at Stanford. And let us go straight to Francis Ashcroft's talk.
Okay, can you see this?
We can.
Excellent. So first I have to apologize because I'm not an epithelial physiologist. I'm interested in how glucose stimulates insulin secretion from pancreatic beta cells and what goes wrong with this process in diabetes. And that's what I've been asked to talk to you about today. So I hope there's some relevance. In some ways, the beta cell is more of an honorary neuron than an honorary epithelial cell. So our studies have focused on a potassium channel known as the ATP-sensitive potassium channel, or KATP channel, because it's regulated by intracellular adenine nucleotides, with ATP inhibiting channel activity and magnesium ADP stimulating it. So when blood glucose levels are low, as shown on the left here, beta cell metabolism and ATP levels are also low, so the KATP channels are open. And this keeps the membrane hyperpolarized and prevents electrical activity, calcium influx, and thus insulin release. Conversely, when blood sugar levels rise, as shown on the right, glucose metabolism generates ATP, which binds to the channel and causes it to close, thereby triggering membrane depolarization, calcium influx, and insulin secretion. Now, the KATP channel is really important because it plays a key role in insulin secretion, and consequently its impaired metabolic regulation results in diabetes. And this can happen either because of gain of function mutations in the channel that render it insensitive to ATP, or which happens in neonatal diabetes, or because of impaired metabolism, which fails to generate sufficient ATP to close the channel, which is what we hypothesize happens in type 2 diabetes. And we've explored how activating mutations in KATP channel genes cause neonatal diabetes across many levels, from the channel to the beta cell islet and whole organism, both mouse and human. And what we've found is that activating mutations act by reducing the ability of ATP to shut the channel, and consequently glucose fails to cause membrane depolarization, calcium influx, and insulin release. The beta cells essentially switched off, and it's unable to respond to glucose, thereby causing neonatal diabetes. Now, it was originally assumed that neonatal diabetes patients had an unusually early onset form of type 1 diabetes, and they were therefore treated with insulin injections, as it was thought they had no functional beta cells. But our work suggested that the reason for the lack of insulin secretion was because the KATP channels failed in response to glucose. And this suggested it might be possible to bypass the metabolic steps with a drug that such as the sulfonylureas, which bind directly to the channel and cause them to close. And these drugs had been used for over 20 years, well, actually 50 years, to treat type 2 diabetes, but hadn't been tested in neonatal diabetes. And happily, we found that the sulfonylureas close most mutant channels and stimulate insulin release. And now more than 90% of patients with KATP channel mutations have transferred to drug therapy. And both their clinical condition and their quality of life improve, as you can see here from some of their comments. However, neonatal diabetes is a really rare disease, and it only affects a few thousand people. The vast majority of people, patients, all 4.5 million of them, have type 2 diabetes. And this is a progressive disease that starts with impaired glucose tolerance and progresses to diabetes as the beta cells gradually fail. And by the time patients are diagnosed, as indicated by the vertical line, it's estimated that only 50% of beta cell function remains. So a key question is, why do the beta cells fail, and why does it happen gradually? And there's accumulating evidence that this is because elevated blood glucose levels impair beta cell function by gradually reducing insulin content and impairing metabolism, which will lower ATP production and reduce KATP channel closure and thereby insulin secretion. And we engineered a mouse in which we can rapidly switch on diabetes, which enables us to look at the effect of hyperglycemia in vivo and look at the time course of it. And here you see islets stained for insulin from control mice, from mice that have been diabetic for 4 weeks, and for mice that have been treated with insulin immediately after diabetes induction. And you can see that diabetes causes a massive loss of insulin content, and that's prevented by insulin therapy. And the reduction in insulin content is caused by a decrease in insulin gene expression, as you can see in the lower right. We also examined changes in mitochondrial metabolism in the beta cell by measuring NADH. By measuring NADH generated by the TCA cycle, by measuring the mitochondrial membrane potential, oxygen consumption, and ATP. And we found all of them were impaired in the diabetic islets. And TCA cycle proteins and OXPHOS proteins were also downregulated. So our results and those of others suggest that chronic hyperglycemia drives the diabetes progression. We suggest that a number of factors such as insulin resistance, age, pregnancy, or a genetic predisposition may produce a small rise in blood glucose, but we don't know which of these are the most important. And what this does is cause a mild glucose intolerance and produces a small rise in blood glucose that may persist for many years. And the problem is that even mild hyperglycemia causes gene changes that eventually impair beta cell metabolism and reduce insulin secretion even further. And this exacerbates the hyperglycemia, raising blood glucose even more and producing a vicious cycle that underlies the progression to frank diabetes. And preventing this cycle may, of course, be of therapeutic value. And as I see it, the key questions that now face the field are, is it ATP? Is it the ATP that causes the channel closure mitochondrially generated? I mean, this is the current dogma, but it's recently been challenged. What, most importantly, drives the metabolic programming in diabetes? And how can we prevent it and thereby slow diabetes progression? Are the same pathways involved in changing the insulin gene expression as for the metabolic genes? And where are the metabolites and metabolic enzymes located in the cell? Are they found in metabolic complexes? Are they stable? Do they move around? Now, we face many challenges in studying this, some of which are unique to the beta cell and others that might apply to many other tissue. So which is the best system to study? There's no ideal system for insulin secretion. All of them are flawed because the cell lines don't secrete insulin in response to glucose sometimes, or their efficacy may change with passage number. Metabolism may change during the time it takes to isolate islets. Islets are often cultured overnight to facilitate their recovery from the stress of isolation. But what glucose concentration should we culture them at? Is it valid to study metabolism in islets anyway, given the lack of vascularization, which means that the center could become hypoxic? How closely does a diabetic islet resemble a cell line cultured at high glucose, given the timescales involved are so very different? How do we change one metabolic parameter at a time to look at its individual effect? That's not easy with metabolism because they will change. How do we measure metabolic flux rather than metabolite concentrations? How can we measure metabolism better in single cells, in tissues, or in vivo? And how do we target probes to specific tissues, particularly in vivo or in the clinic? And of course, how closely do rodent beta cells resemble humans? So I've made a wish list of what we would like, tools to help us this. The first one is we need tools to measure metabolism in real time without disturbing it. So we need sensors for different metabolites. We need caged metabolites, membrane permeant substrates and metabolites, and more specific inhibitors. And we need probes to do this in vitro, in islets, and in single cells at high time resolution. And we also need probes to measure metabolism in vivo. And we need to be able to target them to beta cells and to have sufficient spatial resolution. And we also need ways to measure metabolism and beta cell mass in clinical practice. So finally, let me leave you with a list of left field things to think about. What glucose concentration are you using in your experiments? It may have a big effect on cell function. Many tissue culture media are very high in glucose. RPMI is 20 millimolar, DMEM can be 25 millimolar. These are diabetic levels. It's very odd that some insulin secreting cell lines are cultured at 25 millimolar glucose. And it's also strange that brain slice recordings are often made in 10 millimolar glucose when the CSF is only around 1 to 2 millimolar. And some salt solutions lack any metabolic substrate at all, so cells may be starving or have to switch their metabolism. It's also worth noting, I think, that quite a lot of biochemical experiments have been done in sodium buffers, not potassium buffers, which is not what's found in the cell. Do we need a better model than the mouse? The mouse lemur is a small, rapidly breeding primate that's been suggested as a better option, but it still needs a lot of development. And finally, can we learn from other organisms? Albert Crow of epithelial fame once said, for many problems, there's an animal on which it can be most conveniently studied. And I've always been fascinated by the fact that hummingbirds have blood glucose levels of 30 to 40 millimolar, which would put a human in a diabetic coma, and why, although they live a long time, they don't develop any diabetic complications. And how naked mole rats survive without oxygen for many minutes by switching to fructose metabolism. So in summary, metabolism is important in all cells and integrates across many scales, and I'm sure it's something that you need to consider also in epithelia. Thank you.
Thank you, Frances. That was terrific and raised a number of questions, as you point out, that extend not only to the endocrine system, but also to epithelia as well, especially those relating to metabolism and how to measure it. So at this point, I'd like to ask Allison to share her screen and have Allison Zajac begin her talk.
Okay, great. So yeah, I'm Allison. I'm a postdoc in the Horne-Badovinac lab, and in our group, we're going through scales. So now we're moving from ion channels to looking at some behavior that happens inside epithelial cells. So I work on kinesin-directed secretion and its role in shaping the basement membrane. So the basement membrane is a sheet-like extracellular matrix that lines the basal surface of all epithelia. I pseudo-colored a couple in cyan here, so you can see the sheet-like nature essentially wrapping these different tissues. It plays many important roles in development. It provides mechanical strength to tissue. It's a place for cells to adhere, and it's a reservoir of growth factors that can impact signaling. So despite the importance of basement membranes, we actually know very little at the cell biological level about how cells secrete and assemble these matrices. This really requires a developmental system. And so I'm using a Drosophila model called the egg chamber. So here you're looking at a follicular epithelium. The follicular epithelium makes all major basement membrane proteins. It surrounds the outside of this tissue. And this is really convenient because it assembles its basement membrane, which I've labeled here in cyan, on the exterior. And so we can take this out and image the process of secretion and basement membrane assembly live. So I want to switch to imaging along the basal surface so I can show you what that looks like. So you can see the basement membrane in cyan and the cells are outlined in magenta here. As I play this movie, you can see that one of the unique things about the way this basement membrane forms is that the cells are collectively migrating along it while they're secreting new basement membrane protein. So just so we're on the same page, this is an egg-shaped structure. So the cells are migrating around and around, laying down many new layers of basement membrane. If you look at the collagen channel only, you can see that the basement membrane also has this aligned fibrillar pattern. And we do know that secretion has to be coupled to this migration in order to form this pattern. And so previous work from our lab has shown that the way this happens is a portion of new basement membrane proteins are sent to the lateral domain where they assemble into these fibrils in between cells. And then in the movie, you can see as the cells are migrating, these will attach to the underlying basement membrane and be pulled out and aligned with the direction of migration. And so this is how coupling these two processes can allow you to form a specific structure in the extracellular matrix. The role of the basement membrane in this tissue is to act as what's called a molecular corset. So the egg chamber will grow dramatically over the course of development, and the basement membrane allows it to elongate like you see in this normal egg. And you can see how dramatic the effect is when you don't have this very important basement membrane in the mutant egg chamber. So what I'm really interested in is at the cell scale, how does a polarized epithelial cell that's also migrating know where to send these proteins to correctly assemble this structure? So I just want to zoom in on a single cell for a second. What we already know about this process is that new basement membrane proteins are sent to a RAB10 and RAB8 dependent secretory pathway that's sent to the basolateral domain. But the basolateral domain is really quite large. And so we wondered if there wasn't an additional regulatory step to target secretion more tightly to where it needs to form. So how could you define a subdomain within these larger cortical domains? Well, epithelial cells all have a polarized microtubule array, and we actually know already from mammalian cells that microtubules are involved in basement membrane secretion. And so we thought that transport along this network to microtubule plus ends might be a way of spatially targeting secretion. So I did an RNAi screen and I identified two kinesins required for this process. The kinesin 3 family member, KHC73, is plays the major role, and it works along with the sole kinesin 1 in Drosophila. So I'll show you that kinesins do impact the structure of the basement membrane, and then we'll move inside the cell to see how. So if we just look at the basement membranes in these mutant egg chambers, you can see that even collagen that does reach the basement membrane is incorrectly assembled into these abnormal structures. And if we move away from the basement membrane, even more dramatically, we can see that when you don't have kinesin transport, collagen ectopically forms these cable-like structures that run in between the lateral domains of cells. And so what we think these phenotypes mean is that under normal conditions, the kinesins transport secretory vesicles towards the basal surface prior to secretion. And that when you don't have this, you've expanded the secretion site. So you now have collagen all along the lateral domain that's forming ectopic networks, and even the networks that are transferred to the underlying basement membrane are also abnormally formed. So now I want to move inside the cell and give you some evidence that kinesins are actually doing a transport function in order to cause these basement membrane defects. But first, I want to introduce you to a cartoon I'm going to use from now on. Because these cells do have apical basal polarity and are also migrating, I'll also be referring to the leading and trailing edge of each cell. And I just want to remind you that RAB10 is marking these basement membrane secretory compartments. So the first thing that we wanted to do was look at where RAB10 is localized to get an idea of where secretion might be occurring spatially in these cells. So I'm imaging just along the basal surface, and you can see RAB10 is dynamically appearing at the basal surface and accumulating specifically at the trailing edges of these cells. And this is exciting to us because this suggests that RAB10 is localizing to a place where it's against the lateral domain, but only the basalmost region, which would allow these fibrils to form near the underlying basement membrane. So the next thing we wanted to understand is how could a microtubule motor take you to this very specific location? So how are the microtubules organized? So we know from work from many labs that follicle cells have two polarized microtubule arrays. Like all epithelia, microtubule plus tips grow towards the basal surface. But as the cells are migrating, microtubule plus tips also grow towards the trailing edges of the cells, leading to an overall enrichment of microtubule plus ends in the same region where RAB10 accumulates. But how could a kinesin navigate both of these networks? So I wanted to look at the actual microtubules in 3D. So I'm showing you a single cell with the microtubules along the basal surface. The green box is around the leading edge of the cell. And I constructed a 3D projection to show you how these two microtubule networks are connected. So you can see that the microtubules that are fading from view are actually bending and integrating into the apical basal network. So there is an interconnection that a kinesin motor could potentially use. And so this microtubule organization is exciting to us because it suggests it could facilitate transport both towards the basal surface and also towards the trailing edge of cells. So finally, we wanted to see, do the kinesins that we identified as affecting basement membrane actually affect what we think is the basement membrane secretory compartment? So I looked in the KHC73 mutant animal. So looking at the basal surface again, you can see in tissue lacking KHC73 that it's still migrating, so that's not the issue, but that much less Rab10 is appearing at the basal surface and accumulating at the trailing edges of cells, showing that KHC73 is necessary for both the secretory compartment and the basement membrane. And so we think that kinesins are important for directing the site of basement membrane protein secretion. I just want to take a step back and point out, I'm working in a very specific system, but a polarized microtubule array is common to all epithelia. And so we think that this might be more generally relevant to the way basement membranes are built. But tissue context, of course, matters. So in this migrating epithelium, we think that this unique microtubule array allows kinesin transport to bring vesicles to a specific location that allows them to fuse with just a small region of the lateral domain, which allows these fibril-like structures to form in a region where they can easily be transferred to the basal surface. In the last couple of minutes, I just want to go over some of my wish lists that we were asked to give to Janelia. So I picked this system specifically because there's really nice imaging at the basal surface of the tissue. But of course, there's so many questions we can't answer by only imaging one plane. And so I hope that there's continued development of really rapid live imaging and analysis, and also EMs. There's a lot of interconnection between these migrating cells as well as the overlying germline that I didn't talk about. In parallel, I think one of the things that I really don't understand is what are the contents of all of these different secretory pathways. So we, for example, know the Rab involved, but there's many different basement membrane proteins. So are they all traveling together? We really only look at the core ones that are tagged. And specifically in different cell types, how are different secretory pathways reorganized? For example, these cells are trying to coordinate a collective migration as well as this secretion. So is there kind of redirecting different secretory pathways spatially? However, it's really challenging to think about how you would purify a very transitory compartment like a secretory compartment. So I just wanted to mention one thing that we noticed about the Drosophila secretory pathway that suggested that maybe this would be an interesting model organism to focus on for trying to isolate these types of transitory compartments. So Ben Glick's lab developed a model secretory cargo called S-cargo for use originally that we worked with him to bring into Drosophila. So the way this works is the cargo is originally trapped in the ER, and then you add a drug and you release a pulse of cargo that moves in a synchronized way through the Golgi and into the secretory system. And you're looking at a movie of that here, where in cyan, you can see the cargo that originally is co-localized with the Golgi, and then you see a burst of secretory vesicles forming. And what was exciting to me is that this is a very, very tight wave. So if you compare the same cargo in a mammalian cell to Drosophila, you can see how much tighter the kinetics are for Drosophila, which indicates that there's a time at which this cargo is really predominantly in a post-Golgi secretory compartment. And I'm wondering if there's a way that we can build on this to engineer perhaps the model cargo like S-cargo or something else with the sorting motif, so it will go to types of secretory compartments we care about and bring along a biotinylation enzyme or other way of tagging the proteins that are all traveling together and allow us to start to build up a better idea of what all the different parts are in specific types of epithelial cells that allow them to build things like basement membranes and build other types of structures during their development. So with that, I just want to thank my lab, in particular my advisor, Sally Horne-Badovinac, and Jason Kassler in the Glick lab, who developed all of the secretory cargo, and of course, the fly community that shared a number of reagents over the years. Thank you.
Thanks, Allison. That was terrific. Let's now move on to Helen McNeill. So if you could share your screen and start your talk.
All right. Can you see that? Okay. Yeah, I'm going to tell you about work in my lab and how that leads into the sort of bigger field questions that I hope Janelia will be able to help answer. So my lab is interested in understanding how fat cadherins, these enormous cell adhesion molecules, regulate organ size control and planar polarity. So my first thought was, okay, how do I fit into this 4D physiology meeting? Because I'm not actually working on physiology. But it's pretty clear that organ size, having the appropriate organ size, is really essential to organ patterning and organ functioning. And planar cell polarity, you've already seen beautiful pictures from Lily and Gunnar showing that polarized movement of cilia is essential for function in those organs. So really understanding these basic mechanisms is a challenge and an important one. So specifically, these fat cadherins that I'm obsessed with are huge, so 560 kD cell adhesion molecules that bind to other enormous cell adhesion molecules called doxas. They're massively important for tissue growth, and they work together to organize organ size and planar cell polarity. And this is just some pretty pictures to show, clearly loss of fat leads to a loss of organ size control, and work from another number of people have shown it's through the hippokinase growth control pathway. And this is an example of planar cell polarity in the fly eye and how it still maintains apical basal polarity, but this tissue organization is lost. Okay, so work from a number of labs has worked really hard at trying to understand how these fat cadherins can regulate these two different pathways, planar cell polarity and hippoactivity. And work has shown that genetically, we're able to separate these functions. Another thing that our lab showed not that long ago is that fat cadherins have this additional role in organizing metabolism. And we showed that proteolytic cleavage of the cytoplasmic domain releases a fragment that's imported into mitochondria, stabilizing complex 1. And so the challenge we're facing right now is how do we understand how one molecule can have these very dispersed functions, and how do we analyze it in the context of developing a functional organ? And so the questions are, well, how does fat regulate this PCP, hippo, and signaling metabolism? And is there spatial regulation of function? Temporal regulation. So, we're only looking at single time points or feedback loops. So currently, we're trying to approach this in an integrated fashion. We're using CRISPR to mutate the endogenous locus and do really extensive phenotype, trying to see what very specific mutations will give. We're also tagging endogenous fat with a number of different tags to allow us to isolate complexes in vivo for proteomics. And we're also using approaches like degrade. So a GFP tagged fat, we can degrade it after we induce the degrading ligand. We can also break it with a TEV protease. However, the challenges is that none of these approaches are really fully real-time biological. So when we use the degrade system or the TEV protease, there's still the time for inducing the degradation, inducing the protease, inducing the ligase. We can use proximity ligations on fixed tissues to see if these protein complexes we've identified with our AP mass spec are truly interacting in vivo, but we don't have a sense of their dynamics. And so for me, the questions or the dream tools that I guess was the question we were asked for Janelia Farms is how do we get tools that will allow us to see the development of organ size and tissue patterning in real time. There's a bunch of data from many people showing that mechanical forces in developing organs is important for regulating its patterning and its size and its shape. But the tools we have for measuring mechanical forces are still really very difficult to access. So cutting things, for example, with lasers, isn't really accessible in a living organism in the middle of a fly. Metabolism in real time, like Fran said, is I've pointed out that fat regulates metabolism, but is this something that only happens in extreme circumstances? Is it happening at different times in development? And endocrine signals are essential in regulating organ size. We want to have ways of acutely activating and inhibiting protein function in vivo. So I'm very jealous of tissue culture studies where people are able to very rapidly turn on and turn off protein function. And we're not there yet in being able to do that in either a fly or a mouse models that we're using in the lab. And as I said, we're quite interested in signaling and complex formation in real time. We're able to isolate complexes with AP mass spec, but we can't really see them evolve over time. And sensors that will allow us to see this is really important. I'm just going to take you through a small vignette that shows me why I really need to know in real time and how dynamic and scale oriented this is. So what you can see here is the structure of fat. Here are conserved domains. And here's just a little mini picture of some of the CRISPR mutations we've generated in the endogenous locus. This shows you how conserved some of these regions are. And this is just showing that our idea of phenotyping includes RNA-seq, carefully measuring size and shape, as well as planar polarity. And so one thing that came out of these studies, which I think is a really fundamental question that would be wonderful if Janelia also approached, is how bilateral symmetry is so precisely measured and set up. And so in fly, for example, the left and the right wings are almost exactly the same size. And that's essential for its function because it can't fly properly if they're not matched. And work from another number of labs, largely in Drosophila, but also in mice, has shown that inter-organ communication is needed to precisely define organ size in this way too. And so I don't really have time to go in detail about this, but work from a large number of labs has shown that insulin signaling works with neuronal central synapses and hormonal signals to help coordinate this left and right size of the wings. And so the one reason that I got very excited about this is when we took our very precise mutations that removed actually only a 10 amino acid portion of fat, which is well conserved in humans, what we see is the left and right wing sizes are no longer coordinated. And so to me, this is a truly fundamental question of cell biology and physiology is understanding how this very precise organ size control is coordinated in vivo. And that's why I had as my wish list all these live imaging, because this we're able to very precisely measure those wings to see that there's this lack of coordination in the adult. But we have no way of seeing at this moment really all these different pathways that we know fat is regulated in. We have no way of really understanding how they're precisely coordinated during development to get this beautiful form and function. And so, that's actually the main things I want to say. The only things I'd like to, one more thing. This protein complex formation in real time is also really important. We know that fat is regulating the Hippo pathway. How are we exactly measuring when it's going on? We know that it's regulating metabolism, but is this something that feeds back into these other pathways? And without really tight live time control in a living organism, we can't know that. And so I guess I just want to end by acknowledging the people who did this. The CRISPR analysis was done by a fabulous research assistant, EQ. This actually first an undergrad and now a graduate student, Megan Glazer, has been studying how this left-right organ coordination is occurring. And Juanita has been actually measuring that. And we've also discovered links between fluctuating asymmetry and regeneration. And this is the group. And this is my funding, which I'm also grateful for. Thank you.
That was great, Helen. Thank you very much. So So now the last talk of this session is by Anne Eichmann. So Anne, if you could start sharing your screen.
Gotta unmute myself first. All right, very good. Okay, so thanks very much for having me at this exciting meeting. I work on endothelial cells. And so whichever other cell in the body you might be studying, there's an endothelial cell and a blood vessel sitting right next to it. The blood vessels supply and deliver whatever that other cell needs throughout its life. And so we cannot model blood vessel growth or interactions with organ-specific cells in a culture dish. And so we understand very little about endothelial organ communication. But there is ample evidence that endothelium dysfunction causes diseases in all organs. So there's a clear need to better understand how endothelials communicate with organs to promote health. So we have 4 types of vessels in the body. There are arteries, capillaries, and veins that are connected to the heart, and they pump the blood around the tissue. And at the arterial pole of the capillary, arterial blood pressure drives blood, fluids, and nutriments into tissue. So here's where organ exchanges of oxygen and nutriments occur. And then waste products and excess fluid are returned by the venous and lymphatic circulation back into the heart. And so lymphatic vessels are an open-ended circuit. Their capillaries lie in tissues and they take up fluids and macromolecules and then return them to the venous circulation. And along this path are interspersed lymph nodes where immune cells that are also taken up by lymphatics will be filtered, and so immune response will be communicated. So the system is designed to balance metabolic supply with demand, and simultaneously it mediates immune surveillance as well, and this occurs in all organs. And so the principal characteristics of blood vessels are that they form these hierarchically branched and highly ramified tubes. They have a very simple structure with an inner endothelial lining and an outer lining composed of mural cells. And these adapt to vessel size, with the smallest capillaries only having pericytes, so small mural cells, and larger vessels having additional layers of smooth muscle cells. The system is huge. It covers 60,000 miles if you take all the capillaries and you put them end to end in a single human body. And if you take all the endothelial cells and lay them next to each other, they can cover the surface of a tennis court. So my lab mainly studies vascular development, and I show here pictures of vasculature and chick embryos or zebrafish or mouse organs that we study. And you can appreciate how complicated these networks are already early on in life. And you can also appreciate that they look different between different organisms, but in each individual organism, they form in a very stereotyped manner. And so we believe that by understanding the mechanisms, how this network grows and functions, we will ultimately be able to grow blood vessels for regenerative purposes or stop their growth in unwanted situations of cancer. And so essential functions of the vasculature are, first of all, they have to grow in order to sustain development of new tissues. And this occurs in embryonic development, but also in organ growth and in situations such as wound healing and pregnancy. And in normal adult tissues, endothelial cells do not divide. Their average lifespan is 30 years, but any cell can be activated to divide quickly if a wound occurs. And this process doesn't appear to require any specific stem cells. So once growth has occurred, the system optimizes by remodeling into a perfectly functional system that is able to sustain circulation. And during that entire process, vessels exchange tissue metabolites by forming a semi-permeable barrier that supplies every cell, tissue, and organ in an appropriate manner. And so I wanted to give you 3 vignettes in my talk today, focusing on vascular growth, vascular remodeling, and barrier formation. So regarding vascular growth, we and others have identified some of the mechanisms that regulate vessel growth. And this is a process triggered mainly by the lack of tissue oxygen or hypoxia. And hypoxia will trigger release of factors such as VEGF, which allow these individual endothelial cells to break away from the capillary wall and form these angiogenic tip cells. And tip cells look different from all the other endothelial cells in that they are not luminized, but they extend these filopodia, and these will sense the growth factor VEGF or other factors in the extracellular matrix and make these tip cells. And other cells behind the tip cells will form the vascular lumen, and they will deliver blood and relieve tissue hypoxia and promote vessel quiescence. And these cells use Notch and BMP to become different from angiogenic tip cells. So overall, sprouting angiogenesis involves a division of labor between tip cells that sprout and follow stalk cells that form a lumen and transport the blood. And so my lab started with the observation many years ago that these angiogenic tip cells actually look somewhat similar to axonal growth cones, and they also express shared signaling molecules. And we analyzed over the years several of these molecules and identified some potent regulators of vessel growth that are attractive targets to guide vessels towards an ischemic area or away from a growing tumor. So now let's turn to vascular remodeling. Here I'm showing you 2 chicken embryos with developing vessels. This is an early embryo that has already developed a vascular plexus, but its heart has not started beating. And this is an embryo just 24 hours after the heart started beating and blood flows. And you can see that the circulatory system has remodeled into a much larger system with distinguishable arteries, capillaries, and veins. And so this process depends on the presence of blood flow. It is very fast and lethal if defective, and it involves as many as 50 genes, and we don't understand this very well, but it occurs not only during embryonic development, but also during organ growth. And some recent data have highlighted a very surprising and interesting endothelial cell behavior during remodeling, and that is migration of endothelial cells against the direction of blood flow. And I show this to you here in a mouse model, in a mouse retina, where we use a specific Cree line to lineage trace endothelial tip cells. So this ESM1 is a tip cell specific marker. And if we use its promoter to turn on a fluorescent reporter, you can see that in the beginning, only tip cells are labeled. But 24 to 48 hours later, these tip cells have multiplied and their progeny has moved against the direction of blood flow into the forming arteries here. And so this flow migration coupling process occurs in the absence of any vascular leak and appears to be a major mode of vessel remodeling and artery growth. And we and many other people in the field are interested in finding out how that works at the molecular level. And so finally, I want to turn to endothelial barrier function, which is perhaps the most interesting process of all. And so we know that endothelial cells control transport of everything in the bloodstream, and they do so by forming a selectively permeable barrier in an organ-specific manner. And so the brain needs a tightly controlled milieu to allow neuronal firing, and it has a very tight blood-brain barrier that strictly limits exchanges between the blood and the brain. Other organs, such as the lung, liver, and kidney, are specialized for exchanges of gases, nutriments, and waste products, respectively, and they form a less tight barrier with endothelial fenestrations. And finally, lymphatic vessels are specialized for uptake of fluids and nutriments, and they were thought not to have any barrier at all, even though I will show you that is not true. And overall, we know very little about how these barriers are formed and how they can be manipulated for therapeutic purposes, and understanding this really holds the key to understanding organ physiology. And with the advent of single-cell RNA sequencing, we've already learned that endothelium is super heterogeneous between these organs. So we are on the way to take some giant steps towards a molecular understanding of endothelial barrier function in organs. Now, our entry into this problem came from a study where we knocked out 2 receptors, neuropilin-1 and FLT1, in endothelial cells in an inducible manner in adult mice. And when we put these mice on high-fat diet, you can appreciate that the mutant mice didn't gain any weight at all, even though they ate as much as their littermate controls and they had similar energy expenditure. And the reason for this phenotype was these mice had deficient plasma lipid uptake, and that occurred through a defect in the lymphatic endothelial cells in the gut. And so you know that plasma lipids are synthesized by the enterocytes in the intestinal epithelium into large particles called chylomicrons. And these are too large to squeeze into the blood vessels. Instead, they enter the lymphatic vessels in the center of the intestinal villi through these open junctions between endothelial cells called button junctions. And in our mutants, these junctions had closed, and this created a barrier that prevented plasma lipid uptake and led to lipid excretion in the feces. So the two take-home messages here are that a small change in lymphatic junctions can make a huge change in whole body metabolism, and that the lack of barrier in these button junctions is by no means passive, but requires an active signaling process downstream of neuropilin-1 and FLT1. And I'm a little over time, but because Janelia likes the brain, I wanted to end my talk with a few words about the blood-brain barrier. So you know this is a complicated structure. It involves endothelial cells holding hands very tightly, numerous pericytes that prevent transcytosis, as well as astrocytes, the basal lamina, and a whole bunch of transporter proteins. And the net result is that if you inject a dye into the peripheral circulation, it will go everywhere except into the brain. We made a mutant mouse, this is unpublished work, of a receptor which is highly expressed in brain endothelial cells. And if we knock out this receptor in the adult mice, we get leakage of injected dyes into the brain, whereas in controls, the tracer remains within the blood vessels. And this leakage is size and region selective in the brain, and we're trying to get a mechanistic understanding of how this develops. But just to finish, you know that homeostasis in the brain is regulated by the influx, which is restricted by the blood-brain barrier, and by the meningeals lymphatic vessels that surround the brain and that regulate efflux and brain clearance. And in situations such as aging, hypertension, or hyperlipidemia, both a leaky blood-brain barrier and dysfunctional lymphatic vessels contribute to neuroinflammation, macromolecule accumulation, and ultimately neurodegeneration. So my pitch is perhaps Janelia can help us better understand how blood vessels talk to brain and other cells and vice versa. And I thank you. And I think as a group, we will be ready to take your questions. Sorry, I was a little over time.
That was great, Anne. So at this point, let's turn it over to Maha and Manu, who are going to lead the discussion.
Perhaps the best thing is to first have questions, which basically focus on the individual talks before we open it up broadly. Is that okay, Manu, and with the others?
Yeah, I think that would be good. And if you can all type your questions either in Q&A or in the panelist chats. I only see 1 or 2 questions so far. I'm sure everybody has lots of directed. We'll do that maybe for the first 10 minutes.
So a couple of our panelists have questions, so I'm going to bring them on, and then maybe we can get the discussion started there.
Yeah, I think while we're starting, one question I see in the Q&A from Lopez Share, this is for you, Helen. Any evidence that HSP90 or any other chaperones buffers variability in organ size within individual flies?
I don't think anyone's done the analysis of, so the variability of left-right wing asymmetry is called fluctuating asymmetry. And there's been a bunch of mutants that have been identified, not to my knowledge for that one. There can always be something new. This is a new field for me, but it would make sense. I can see that. Where the signaling is that allows this isn't really clear yet.
And I think just touching on that same left-right symmetry, I was wondering whether when you showed that remarkable picture of a smaller wing, is that a scaling defect where the wings scale small, or is there one dimension?
Right. So we're really trying, that's a super interesting question that I could have spent an hour on. So basically, so we measured not just wing size, but also the width and the length. And we've actually, I said there's a 10 amino acid mutation, but we've actually reduced it sometimes to as little as 3 amino acids. And what we can see is that we can affect fluctuating asymmetry without necessarily, it's not linked to size, though the same domain can, in slightly larger mutations, affect them both. We're seeing differences in the roundness, let's say. So the proportions do get changed. we also see changes in planar polarity even down to those very small ones. So we're still really struggling with, we haven't been able to cleanly dissect out that aspect. But yeah, there are shape effects that are correlating with the FA2. Yes.
I think there are some questions, maybe just in order: Marino, Zerial, Prabs and Gupta, Ron Whale, and Elaine Fuchs, and then Thomas Lequi.
Yes, thank you. I had a question to Allison, and that is that I would imagine that in this condition, the cells which have to move are secreting, but they're also having to recycle, and probably recycling tubules will, circulate again, additional molecule like a conveyor belt in a way. I was wondering, when you look at that, and it seems to me that this system is also similar to the retina when it forms, where you have the epithelium, they undergo this incredible transformation where the cells have also to move and tumble around. So I was wondering anyway if you had any insight on this turnover of membranes, more than just the secretion.
Yeah, great question. So actually, one of the reasons I brought up that I'm so interested in trying to understand like how the secretory compartment might be remodeled in these cells is because where I showed the RAB10 localizing at the back of the migrating cell, that's also where the adhesions are needing to turn over. And so the microtubules are aligned, kind of pointing to like laying over top of the adhesions, and the RAB10 is sitting on top of that. And if I were to just show you like adhesions and RAB10, you would think that they were tightly coupled. And so we think that probably there's other types of RABs also localized in that location, and that there probably is some coordination between needing to turn over integrins at the back of the cell and also secreting to that area. So there seems like there would be a lot of different membrane turnover right in that region. But at the light microscope level, like everything just kind of looks perfectly co-localized. And so getting at like causality and mechanism is really difficult. So I think like having EM and being able to see what the organization is in that region will be really important. And like one of the things Helen brought up, having acute loss of function, I think would be really essential. It's really difficult to say, like if we perturb basement membrane secretion, for example, there's a massive disruption to the ability of the cells to migrate. And so like, how can we tell how integrin trafficking is different from ECM trafficking? I think in some systems, people have also suggested that integrins are the receptor that sorts certain types of ECM proteins. And so like, they're actually involved in like finding them in the secretory pathway, soluble cargoes, and transporting them. And so I think the way the cell kind of organizes everything in one location is really interesting and also like really complicated to tease out. And then relevance to other systems. I mean, I'm aware of those migrating systems, and so I think it would be interesting to look, but I don't know anything specific.
There you go. Okay. I had a question for Francis and also the panelists in general. I was thinking of whether it's worth pursuing a paradigm of how metabolism informs cellular form, function, and also organ, and basically like shape of organelles within cells. And for example, how much do we know, like for example, in the epithelial villus, like the cells at the base compared to the cells at the tips, they are very different amount of oxygen that they can actually access, right? So is there basically a spatial organization of metabolism along a structure like villus, and whether that actually informs the function of the cells?
That question was to me. I can't answer that because I don't know about the villus. But I do think that, different cells have different metabolism. I have a question about that for Helen, actually, but I do think it's worth considering.
There is actually a very patterned cellular metabolism along the crypt-villus axis. So yeah, it is regional.
So basically it does, but the thing is, other than that, like, because normally we think of metabolism are like the cells have certain functions, and basically depending on them, they perform certain kind of metabolism. But can we actually basically shift the shift that paradigm, that actually metabolism can have also a dominant role in terms of determining the cell's basically function and the form, right? Is that one a way of, we can think, especially in tissues where you have very different kind of microenvironments?
Yeah, no, it can shift in response to nutrients. I mean, you can get a shift in cellular metabolism in response to luminal nutrients in the intestine and change the depth of the crypts, the proliferative rates of the progenitors. So yeah, certainly shifts in metabolism
perhaps best known shifts due to dietary changes can certainly change the form as a result.
They can change the form and the fate, right? With regards to changing succinate, alpha-ketoglutarate ratios and the like, and affecting in all a host of other things that can affect the epigenetics. But
maybe we should continue because there are a large number of questions, if you don't mind. So Ron, and then Thomas, Elaine, and after that, I've started to lose track. I don't know, Manu, if you've been keeping track.
Yeah, G2 and then Francis again. Ron.
That's a long list there. Well, first of all, I just wanted to say thanks to Allison for a great talk. We worked on that kinesin like 10 years ago, so it was really exciting to see all the progress that you've made on it and all kinds of new understanding. So it'd be fun to talk to you more about that. But the question I have right now is for Helen, and it's kind of a big picture question about organ size and symmetry. So I was really interested in this symmetry problem, but then there's this organ size problem, which is as old as simple organisms like planarians up to humans, about how you get the right proportions between different organs in the body plan. And my question is, do you think that so these are really big questions of biology. Do you think the problem of relative organ size and symmetry are one and the same kind of problem in terms of Maha saying no? So anyway, well, maybe both of you could answer that question then.
I'll hand wave. Our own experiments suggest not, because we've done this. I mean, I only talked about one region, region D, but we're able actually to find domains in fat which can affect the organ size but aren't affecting the symmetry. So at least under that one condition, we can separate it. I mean, I think ultimately things have to be integrated together, and that, of course, that's a weakness of our approach, right? I mean, we take out this region and we see sort of what the net effect of loss of that region is. We're not seeing the total integration. So, I think that's like yet another level of seeing perhaps there are feedback loops that we're not affecting through those static mutations. And again, like Allison said, just trying to see, what the acute effects of changes, to me, has become like an obsession, like a crazy obsession. Yeah.
I'm going to wait because I think there's a bigger question over there, Ron, that you raised, which I think perhaps could be part of this larger question of how to characterize, how to quantify shape and size variability. So that's a larger question. Perhaps I'll leave it till later, if that's okay, because we want to basically get everybody who's got their hands up to get a chance before we switch in about 7 or 8 minutes to broader discussion. So Thomas, Elaine, G2, at least that's what I recall.
Yeah, so my question, Helen, is for you, and that actually relates to Ron's question before, and that is that of the tissue specificity of the fluctuating asymmetry problems that you reported in the wing. Do you see the same in the legs?
Yeah.
So I don't know how you did the experiment, and the related questions is that of developmental timing. So usually it's unknown whether there are endogenous fluctuations that are sort of controlled at some developmental time. And if there is an asymmetry, then there's delay so that you catch up. So have you looked at that? Can you tell us about how you couple, tissue-specific asymmetry with developmental timing and sort of the global control mechanism?
Yeah. So those are great questions, and most of the work in this field has not been done by us. Absolutely, developmental timing is linked with, let's say, the DILPs, right? Obviously, or when you have damage, right, you slow down and mess up that DILP signaling, you'll mess up fluctuating asymmetry. But however, we actually spend quite a lot of time looking at the D mutants, and at least during classic, under normal food conditions, pupation is exactly the same. So we don't see. So that question, it's not at least within, absolutely the same, at least within 3 hours. We didn't see anything notable with lots and lots of experiments and other controls. In terms of organs, these are whole animal CRISPR mutants. So it's easier to measure the wing because, as you know, it's a flat surface and it's, basically a 2D structure, so you can pick up subtle changes. But, just anecdotally, we didn't quantitate it, but the legs can sometimes be different. The eye sizes can be different. Sometimes some will get stuck in the pupae where we're not exactly sure what's going on with those. So we think it's a whole body effect. Work from other people like Pierre and Maria have shown that, you can damage, and other people, you can damage one organ and it'll affect the growth of the other. So it's super interesting, all these feedback loops. And I think, it's a really important question that's hard to get at.
So Anne, this question is for you. And I was fascinated by your comments about how hypoxia can induce the sprouting of blood vessels. And I get why you would want to do that. And I'm wondering what happens in a wound where you've severed both the blood vessels and the lymphatics. And now you want to trigger, presumably you're not triggering lymphatic sprouting when you have hypoxic conditions, but maybe I'm wrong about that. But then I would think in wounding, you've got to do both. And I just wonder if the mechanisms are known for that and what you know about that.
Yeah, thanks for the question. And it's a great question. So for lymphatics, we know very little about what promotes their growth. And the idea is that we know it's not hypoxia. Hypoxia does not induce lymphatic vessels to sprout. But what are the factors? Is it interstitial fluid pressure? Is it the arrival of immune cells that carry, growth factors for lymphatic endothelial cells or such things? And how is the growth of both of these systems coordinated? It's a great question, and we don't have a good answer for that yet.
It just strikes me that in disease states, there's so many conditions, whether it's cancer or whether it's aging or whatever, where lymphedema plays a huge role. And it seems like if you could figure that out, that would be a big plus.
Yeah, absolutely. Is a great area for future research. Thank you for the question.
And I had a sort of a similar question to Elaine's, but in somewhat more mechanistic terms. I mean, how is the ischemia sensed and then relayed so that the sprouting angiogenesis moves towards and opens up new vessels? I mean, that would be one question. And then clearly during wound healing, is the same machinery used? I mean, during arterial, for example, regeneration in ischemic heart and things like that, do you get the same sort of mechanism operating?
Right. So there's several steps that occur. The first step for blood vessel growth is, release of factors usually produced by a hypoxic stimulus, but it doesn't only have to be a hypoxic stimulus. It could be a drop in pH or some other factor. And then once that occurs, a sprout is formed. And at some point that sprout becomes perfused, and that triggers this interesting behavior of the cells where they start migrating against the direction of blood flow. So when you think you've got to build an artery, right? How do you add new cells to that artery? And you do that by making them come from the capillary plexus, and they are attracted by the arterial blood flow and migrate backwards through, an entirely already perfused vessels. And that's really fascinating process that mechanistically we don't really understand how it does that, and how it does that without having leakage of blood and hemorrhage into the tissue. So,
and during the regeneration, I mean, is there when, for example, in ischemic heart condition where
yes, so the data show that it's a similar type of mechanism, and involves a lot of different endothelial behaviors, because once they are in the artery, they stop proliferating, right? There's a lot of oxygen there, and so they don't proliferate, right? So you can't easily make an arterial cell make a new cell. So the way the system does it is it calls cells from other areas of the capillary bed, and these cells will grow the arterial branches. So it grows from the outside by adding cells from the outside. I hope that sort of.
Yeah. Fascinating. Yeah, thank you.
So maybe we have two more hands from Francis and Helen, and since you were also speakers, you can also transition and we can initiate into discussion soon. I'm seeing the 12-minute mark.
Well, I wanted to ask a question to Helen, which is about this fascinating cadherin fragment that is imported into the mitochondria and stabilizes complex 1. And I wondered, is the proteolysis regulated, and what does that? And, you know, and is the similar thing found in humans?
Right. So, I mean, those are obviously excellent questions, which I wish I knew the answer to. Is it regulated? Exactly. That's why I want real-time measurement of metabolism to see, because we just looked at null mutants, right? To see the destabilization of complex 1 by Blue Native page. The question of whether it's conserved to humans, that we haven't truly addressed at all. Right now, we have a lot of mouse models that we've, like this CRISPR approach that we've done with flies. We're also taking everything we see in one, we go to the other. So, and we're trying. At this moment, I'd say the only clear data we have is in the fly. The mouse, we have hints, but proteomics in certain tissues shows a fragment, but I mean, we don't, you know, I don't know yet, really.
Thank you.
Oh, I guess my question was for Anne. So those, that was pretty fascinating, the junctional regulation blocking obesity. So is that kind of junctional regulation happening normally? Is this, is this sort of daily or hormonally, or, you know, and I guess also, are there any negative effects? Should we just all like affect that and not eat our Oreos or whatever?
No, it's a great question. Thank you. So we know from studies that analyze the development of these junctions over time that there is before birth, mouse embryos have all their lymphatic junctions in a zippered configuration, and yet they do not develop edema. So this specific type of junction configuration must be compatible with fluid uptake into the lymphatic system, but it's not sufficient for lipid uptake in the lymphatic system. And so the remodeling occurs gradually over postnatal development and is probably triggered by, environmental exposure to antigens, that we ingest or that touch our skin. And so over time, gradually these button junctions open, and there's a molecular feedback mechanism in the intestine that we identified, but there could be others. This could occur in a tissue-specific manner, and it's not fully understood. So experimentally, you can dynamically switch these junctions from a closed to an open state with drugs, and that occurs in the order of a half an hour. It goes really quickly. But then in normal situations, they revert back. So better understanding of these dynamics may give us a lot of new approaches to, manipulate obesity, but also inflammatory conditions in which lymphatics are a big problem. And so we also know from inflammatory studies, when there is inflammation in the lung, for example, this was studied, the lymphatic endothelial cells start growing to make more vessels. And over the initial period of growth, they also revert to these zipper junctions, and that aggravates actually inflammation resolution during the early phase. So that's kind of what we know about this.
Cool, thanks.
Oh, I guess if I could jump in for just one quick question. I thought one thing that came up both in Helen and Anne's talk was this ability to make sort of acute perturbations. And I guess this was a little bit raised in Ron's question as well, as sort of decoupling the sort of phenotypes that you see in a tissue and just normal tissue function versus what happens during development. So, if you make a CRISPR knockout, of course, unfortunately, it could be neonatal lethal in the worst case scenario, and then you have no idea where to even start. So I guess, does anyone have any ideas on how you can really decouple those two, so making an acute perturbation in a living organism, and then studying how sort of function really follows that perturbation?
Mm-hmm. So I can maybe start, Helen, you can chime in later. So in the system we study, most of the mutations I've showed you, if you were to do them in a global knockout system, you would get an embryonic lethal phenotype. And so there's very little to study. Vasculature doesn't form, you get a very a tiny embryo that is already dead. And so the way we do this is with inducible approaches where we deliver a drug and that will trigger our knockout. And even better is if we can do that in a mosaic fashion with just a few cells, and that will allow us to monitor their behavior. But we would like to ultimately be able to manipulate endothelium, say, only in one organ, or say only in that one segment of the lymphatic vasculature, like the lacteal. And that would be insanely helpful to help us understand what's going on just in that place and not on a systemic, without confounding effects of a systemic perturbation. And for this, I think we need a lot more of these single cell RNA sequencing data, and we need to identify markers that will allow us to do that. And we don't know these molecules yet.
So maybe on that, should we switch to broader themes? Unless there, okay, so may I start by basically trying to see something which perhaps everybody thought about, but nobody talked about, which was how do you quantify the variability in phenotypes, whether it's phenotype associated with the pattern in space, whether it's the phenotype associated or the pathophysiological phenotype that Francis Ashcroft talked about, particularly over very long times. I mean, so in other words, given the data, if you have these remarkable 4D images in space and time, how do you measure variability which is normal? How do you measure variability which then borders on pathophysiology, and where are those boundaries? And these are, I think, just as much mathematical and physical questions as they are biological questions, because one has to basically come to terms with where is the variability sitting, on what scale is it sitting? And I want to sort of maybe use this as a way to separate what was a spectacular revolution in the field in bioinformatics, where you had a finite alphabet and you were able to do bioinformatics with that finite alphabet and try to understand how to register and then how to look at differences. How should we be thinking about answering these questions about patterning, questions about size, questions about fluctuating asymmetry, questions about physiology on very, very long times? And I'm just curious about what people think, because if you have that wish list of the tools and the data, and then you have all these remarkable images, what are you going to do next? I have some thoughts, but I'm going to basically leave that as a question. And
yeah, I think, I mean, chiming in on this, I love the analogy to bioinformatics in some sense. And often enough, you can't even compare data from one model system to another model system. You can't even compare sometimes data from one lab's tissue culture to another because of specifications that come about. So I think it's a remarkable call to action in some sense for large-scale data sets for exactly the same sets of systems, possibly even collected in much more centralized. I'm reminded of a recent paper where a couple of machine learning algorithms in microscopy were able to pick which graduate student collected the data. Just literally about, there are certain things about data collection that have to be standardized.
Yeah, and maybe coming back to Ron's question, you know, size and shape are different because shape is relative size. So you got to scale out, remove the overall size, you know, and then whatever is left over is what we call shape. But shape itself is dependent on what dial you use, how much you've tuned your microscope, you know, how much I'm zooming in and how much I'm zooming out. So there is a scale dependence there as well. And so where does one decide, and when does one decide that you have enough? And I want to sort of switch that to also, and, you know, your beautiful images where you showed branching, you showed what happens with the plexus early on. How do you decide what is normal? How do you decide what is pathophysiological? How do you decide, and when do you decide? There's a spatial aspect to it, but there's also a temporal aspect to it. You know, sometimes animals, organisms basically develop slowly. Similarly, in diabetic, I don't really know, you know, how people essentially right now decide where you are along that insulin resistance path. If you have the data, how do you convert that to quantification? I'm sorry, I have a certain hat that I'm wearing now. I apologize.
I mean, and so in the system that we study, we know there is a lot of variability. We know there is variability even between individual mouse strains in, for example, the response to stroke and so on. So variability is a problem in our experiments. And the scale of the system we're studying is, in a single brain is 400 miles of blood vessels. So, clearly this is complicated. And so the way we approach this is to use a simplified model system. For example, this mouse retina preparation I've showed you is a fairly reproducible between individual animals. And it happens in 2D, right? So the initial sprouting is in 2D, and you can see it in the entire organ on one small microscope prep. So that has been very, very helpful for people to get a more quantitative understanding of, what is normal and how things work. But overall, I don't have an answer. I think it's great you bring this up. And when we think about translating all these things to humans, then this becomes a much bigger problem, even because, unlike our mice, they're not all genetically identical. And so their responses vary much more than what we study in experimental animals, right?
I also think the more information you have about really looking at your specimen is really important. So I didn't even touch on this, but one thing that we discussed, to these experiments, you had to do left and right wings, and we analyzed it. Turns out female flies have 10 times as much fluctuating asymmetry as the males, right? We have no idea about that. And if we hadn't sex separated them when we were looking at it, we just would never have picked it up. So, I mean, as the more seemingly non-relevant data you can quantify before you try to analyze it, is they give you surprises.
Yeah. If I can add, this connects to human genetics in a number of ways. There are about 3,000 genes that we recognize are haploinsufficient. If you have half a dose, that's not enough. The consequence of that turns out to be surprisingly variable for some of these genes, with phenotypic expression that ranges from incomplete penetrance. Half the people who get what appears to be a Mendelian mutation have no phenotype, particularly on developmental phenotypes, such as you have to make a particular decision at some point in development, whether you're going to loop a heart tube, for example, and whether you do that properly or not is up for grabs. And thinking about things that you might operationalize, if we know that 50% of a dose is generally insufficient, we don't know how much variability there is at the critical developmental time around that point, right? Do individuals who are haploinsufficient have 50% of the dose, or is it a range between 30% of a dose and 70% of a dose? And does that explain some of the variability? And obviously, if you had good in vivo ability to monitor at these critical developmental times and windows what the actual levels of expression were, you might be able to come up with better predictive models of what's going to happen. And that's just thinking about particular discrete phenotypes. But another aspect that is increasingly apparent is that there's a very high degree of phenotypic variability in the expression of the phenotypes. So there are many component phenotypes that you might get from an individual mutation, but which ones appear in individuals is quite dependent on completely unknown factors. And does that relate to the levels of expression in particular tissues at particular times? But these seem like the kinds of things that you actually, a place like Janelia could make inroads in.
So thank you, Rick. This has been a superb discussion, but being like Rick, something of a renal physiologist, I'm sensitive to the fact that we only have a few-minute break in the entire course of this meeting. So if it's okay with everybody, I'd like to stop the discussion here and ask everybody to be back in 9 minutes or so that we can start the next session. Thanks for a wonderful session.
Helen, if you have.
Still there? Uh-oh.
Oh, I guess this is an optional break, huh? So we can all still chat.
Well, I'm sorry to interrupt this really fun discussion during the break, but I think that we should get started. with the next session. So I am really pleased to introduce the speakers and discussion leaders in session 3, which is entitled Digging Deeper into Epithelial Interfaces in Health and Disease. We will start with a talk from Rick Lifton, professor and president of Rockefeller University, then move on to Elaine Fuchs, professor and HHMI investigator, also at Rockefeller. Then we have a talk from Corey Simpson, a fellow in the Holzbauer Lab at the University of Pennsylvania. And then after that, the discussion will be led by Jennifer Lippincott-Schwartz, senior group leader at Janelia and co-organizer of this workshop, and also Jitu Mayor, professor and director of the NCBS in Bangalore. So right now I'm going to go ahead and turn it over to Rick Lifton.
Thank you. So what I want to do today in the brief time is to just talk a little bit about the role that human genetics can play in understanding complex physiology.
Rick, can I interrupt you for 1 second? Do you want to put your presentation on play mode full screen?
Thank you.
There you go.
So a starting point that I want to make is that rare mutations with large effect identify genotypes that cause phenotypes, and that then provides the opportunity to dissect the causal path that connect these. It obviously can identify mutations that underlie both cell autonomous and non-cell autonomous phenotypes, which I think is particularly relevant to many aspects of physiology and epithelia. And it's particularly helpful for understanding causal elements in integrated organismal physiology. I'll point out that there are 19,000 human genes. We understand the consequence of their mutation in the context of a living human of about 6,000 or 7,000 of these thus far. So there's a lot that remains to be done, and the tools are now very powerful for doing that, with the ability to very inexpensively do exome or genome sequencing. So I will confine my discussions to epithelia, but we've worked in many other systems that are not traditional epithelial systems. One of the phenotypes that we cut our teeth on is blood pressure homeostasis. This is a working model for the trait. It's complicated and passionately argued where the underlying regulators turn out to be. We studied people with the highest and lowest blood pressures on the planet compatible with survival, and demonstrated that the virtually all of the rare mutations with large effect impart their effect by altering sodium and chloride handling along the nephron. And this shows some of the cell types and the syndromes that have mutations in individual genes. For many of these genes, gain-of-function mutations drive blood pressure to the highest levels compatible with survival. Loss-of-function mutations do the opposite, and we figured out the mechanisms by which many of these operate. I'll give one example of where new physiology was uncovered by human genetics, completely unknown previously. The regulation of aldosterone production is regulated by two independent mechanisms. One is volume depletion that leads to activation of the renin-angiotensin system, and this produces aldosterone, which in this context tells the kidney to hang on to sodium and chloride. But it's also been well recognized that hyperkalemia is a direct secretagogue for aldosterone, and in this context promotes increased potassium secretion. And so the question is, when the kidney sees aldosterone, how does it know which it's supposed to do? So by human genetics, we identified patients who have a confusion in this point. They can only use aldosterone to promote increased salt reabsorption, never to secrete potassium. So these patients have severe hypertension and high serum potassium levels, and the mechanism of this was completely unknown and ultimately led to identification of mutation in 4 different genes, 2 Wnt kinases, and then a ubiquitin ligase based on Cullen 3 and Kelch-like 3. And it turns out that the Wnt kinases are normally bound by Kelch3 and targeted for degradation, and the mutations impair that process. So the question is, what physiology does that then link to? And again, once you know the genotypes and the phenotypes, you can work to understand how that works. And it turns out that the Wnt kinases are regulating a number of key ion transporters in the kidney. Shown at the top is that angiotensin II regulates the phosphorylation of Wnt kinase, which prevents its degradation. Wnt kinase, once it's free, is capable of phosphorylating SPAC, which phosphorylates the sodium chloride cotransporter, which is one of the important determinants of sodium chloride cotransport. It turns out that this influences the activity by which the epithelial sodium transport in the distal part of the nephron. This is an electrogenic step, and electrogenic sodium chloride reabsorption can either be accompanied by potassium secretion or by chloride reabsorption. And regulating that balance obviously is a key determinant as well in regulating the balance between salt reabsorption and potassium secretion. And this provides an unanticipated layer of regulation by which salt reabsorption and potassium secretion are oppositely poised. And it's a key regulatory element by which the kidney balances the need for both. I'll just give a couple of examples also in epithelial physiology where unlikely that these things could have been discovered, or at least hadn't been discovered by physiologic analysis alone. In the upper left corner, you see a typical epithelial barrier with tight junctions, which we've heard about from Michael and others already at this meeting. Renal physiology had suggested in the lower left that in the thick ascending limb of Henle, there is sodium, potassium, and 2 chlorides that come in on the cotransporter NKCC2. Sodium and chloride escape across the basolateral membrane. Potassium is recycled into the lumen, creating a positive lumen, and this provides the electrical driving force for reabsorption of magnesium and calcium, which was believed to be by the paracellular pathway. But mediators of the paracellular pathway were completely unknown. And by positional cloning in families with the rare form of severe magnesium wasting, we identified mutations in a Claudin 16, that are mutated in this disease. And again, this turns out to be tight junction protein at the paracellular barrier. And it is likely that this is a component that regulates the ability to selectively pass cations. There are a number of negative charges at the first extracellular loop shown in yellow, mutations that cause loss of function. We now have structures that have been done by others of parts of these tight junctions and proposed models that might allow for selective passage across the epithelium. This has been applied to many other areas of renal physiology. Shown here is Friedhelm Hildebrandt, with whom we've collaborated for these gene discovery programs. And he has discovered a very large number of genes that regulate the activity of the slit diaphragm in the kidney, which is what prevents the filtration of protein into the urine and is the selective barrier for filtration into the urine from the plasma. And many years ago, the classic mutations in nephron were identified, but Friedhelm has collected patients from around the world with steroid-resistant nephrotic syndrome, spilling a lot of protein in the urine, which leads to ultimately to kidney failure in most of these. And he's identified mutations in many pathways that were completely unanticipated. I would not have guessed that mutations in nucleoporins would give this phenotype, nor that mutations in CoQ biosynthesis or the Cheops complex would be yielding this phenotype. So I think beautiful examples of non-cell autonomous phenotypes that have resulted in very interesting physiology. Similarly, Steve Somlow, working on polycystic kidney disease, a severe and important cause of kidney failure in the human population. These large kidneys shown here with cysts. And a couple of genes were identified that cause this dominant trait, but the mechanism has remained unclear. And by looking in polycystic liver disease, which is a related disorder, obviously, he's identified mutations in pathways that modify proteins along the secretory pathway that have been critical to this trait. And this has led to recognition that what these are doing is they're essential for the maturation and delivery of the gene product PKD1, which is a ciliary protein, to get ultimately delivered there, and that all of the different mutations that cause polycystic kidney and liver disease underlie the need to deliver PKD1 to the cilium, and defects in that pathway ultimately cause this trait. We've done similar things in many other diseases, ranging from developmental abnormalities, hydrocephalus, congenital heart disease, cerebral palsy, autism. And I think it just underscores the point that genetics is a wonderful partner for physiology, and we have an awful lot that remains to be learned. So I'll stop there. Thanks very much.
Thanks a lot. That was a great talk, Rick. Now let's move on to Elaine Fuchs.
There we go. Okay. Can everybody hear me? Is that good?
We can hear you. You want to put your presentation?
Can't hear me?
Yeah. You want to put your presentation in full screen? Can you hear me, Elaine?
Yeah, I can hear you. How about now?
Yeah, we can hear you. You want to, can you put it in full screen?
Now?
It's still not in full screen.
Can't hear me?
We can hear you.
We can hear you.
Can you hear me now?
We can hear you.
You can? Okay, good. Okay, great.
But can you, Elaine, can you hear me? Can you put your presentation in full screen?
Yeah, I can. Okay, sorry about that. Okay, so I thought I'd like to just use these minutes to perhaps give my view or my take on the importance of epithelial biology. And I'll just start with considering the world's major health problems. And in a world with an escalating climate change, contaminated foodstuffs and water, air pollution, allergens, we face increasing risks of cancer, chronic inflammation and allergies, inability to cope with ever-increasing emergence of new pathogens and microbes as evidenced by our current pandemic, but also Ebola, antibiotic resistance, staph infections, and so on and so forth.
I think your slides are not moving.
Elaine, your slides are not advancing. Do you have it on full screen mode on your computer?
Yeah, I do.
Okay.
Let me just go again. And can you hear me?
Yep.
Okay, I'm going to try sharing my screen again, and let's try it one last time. See if that works. It's not working.
No, we can see it, but what we can't, just go into the PowerPoint mode.
Okay, I'm gonna have to, sorry about this. Normally it works without a hitch, but I'm going to open this up again. It said right beforehand that there was something wrong with there was an interruption, so maybe that was it. Let me just try again. Can you guys see this now? Can you, if I share my screen, can you see that? Can't hear me.
No,
It hasn't come
no,
yet, Ellen.
we can't.
This is usually the problem. Do you have two screens?
I think Elaine's Elaine, I think your connection that's fine.
My connection is bad. I'm going to reboot.
That's fine. Elaine, why don't you join us? And right now, let's move on to the next talk, and then we will come back to Elaine after Corey Simpson. So let's move on to Corey Simpson right now, and we'll come back to you, Elaine.
Yeah, exactly.
It looks like Dr. Fuchs, are you able to unshare your screen?
Yeah, I think she's stuck. Let's just give her a minute to sort of log off. There we go.
Okay, can you see my screen now?
Yeah, looks great.
Wonderful. Well, thank you very much for this opportunity to present my postdoc work in Erica's Holzbauer's lab, where I've worked to convince a group of neuronal cell biologists that the epidermis beats out the brain as the more interesting part of the ectoderm. So as a physician scientist in dermatology, I've been trained to recognize how epithelial dysfunction manifests as skin disease, as shown there on the left. And in the lab, I've used cell biological approaches to understand how the epithelial tissues, their constituent cells, and their organelles support the function of the epidermis. In harvesting the power of live microscopy, we can now visualize the dynamic physiology of organelles with the hope that an improved understanding of epithelial physiology will feed back to advance our ability to treat human disease. So the epidermis is a stratified epithelium made of primarily keratinocytes, which undergo a distinct program of differentiation. Proliferative cells in the basal layer regenerate cells in the upper layers that eventually initiate a process called cornification, in which they eliminate their nuclei and organelles to form tightly adherent barrier layers that keep pathogens out and keep water in. And this is essential to terrestrial survival. So my work is focused on how differentiating keratinocytes remodel and eliminate their organelles, as well as how the health of organelles in proliferative keratinocytes is maintained despite numerous stressors at the body's surface, including UV radiation. So during my PhD in Kathy Green's lab, I learned the power of organotypic epidermis built upon Dr. Fuchs's fundamental studies on culturing human keratinocytes. So in this model, fibroblasts are embedded in a collagen matrix within the transwell, and then primary human keratinocytes are seated on top as a monolayer, submerged in a liquid medium. The medium is then removed from the apical side of the cultures, and this simulates construction of a multi-layered epithelium. Conventional imaging of these cultures is done in cross-section using H&E staining, which reveals that cultures recapitulate mature human epidermal histology in a little more than a week. Immunostaining these tissue sections shows the morphology of organelles in a static way, like TOM20 here marking mitochondria, but this is akin to looking at a fossil record without ever observing the live animals. Thus, we coupled this 3D model with live microscopy methods to reveal the dynamic behavior of organelles like mitochondria within the live tissue. So to elucidate the biology of differentiating keratinocytes, we initially focused on mitochondria given their diverse functions in signaling, metabolism, and cell fate decisions. After using a viral vector to tag mitochondria of keratinocytes, we allow them to stratify, then utilize spinning disk confocal microscopy to take Z sections through the live tissue. So while in the lower layers, mitochondria displayed their classical tubular interconnected morphologies, the organelles underwent a dramatic shift in the upper layers where they became stably fragmented and spherical. We used this organotypic model to then dissect the mechanism of this process and to show it was a critical step in epithelial morphogenesis. So we can transduce keratinocytes with a tandem fluorophore, an autophagy reporter targeted to mitochondria. So dual GFP and M-cherry fluorescence was seen in the tubular mitochondria within the lower layers, but quenching of the GFP in spherical mitochondrial remnants in the upper layers suggests that they might be routed to an acidic compartment, as is known to happen in mitophagy, in which mitochondria are degraded in lysosomes. But what serves as a signal to drive this critical step in epidermal differentiation? So in a focused search of mitophagy receptors, we found a protein called NIX that exhibited increasing expression in maturing epidermal cultures, and its tissue localization in the epidermis was correlated with the fragmentation of mitochondria in the upper cell layers. So NICs had been previously shown to have a critical role in the maturation of erythroblasts, which also undergo organelle elimination to become mature red blood cells, leading us to hypothesize that NICs might actually play a parallel role in the epidermis to drive the differentiation of keratinocytes toward a distinct cell fate, but via a conserved mechanism of mitochondrial degradation. So to test this hypothesis, we transgenically expressed GFP-labeled NICs throughout the epidermis using a constitutive promoter. And this was sufficient to induce the fragmentation of mitochondria labeled in green in the left cell compared to a neighboring cell lacking NICs, which exhibits the normal tubular morphology. In the organotypic model, we showed that expanding the normal restricted pattern of NICs expression resulted in premature tissue differentiation, thinning of the epithelium, and precocious cornification. So on the flip side, we demonstrated the necessity of NICs for epidermal morphogenesis by generating NICs knockout epidermis from human keratinocytes in which the NICs gene was disrupted using CRISPR-Cas9 targeting. So depleting NICs expression impaired the keratinocyte's ability to eliminate mitochondria, as shown in the right EM image, in which tubular mitochondria are retained in the uppermost layers, while they're fragmented and rooted into degraded compartments in the control tissue on the left. So these data led us to propose a model in which NICs serves as a signal to trigger the degradation of mitochondria in the upper epidermal layers, initiating the final stages of differentiation. We further define the mechanism by showing that NICs engages the mitochondrial fission machinery via the GTPase DRP1 to induce mitochondrial fission and drive epithelial differentiation. We then could use this model to specifically compromise DRP1 function using a dominant negative mutant. So live imaging of mitochondria, as shown here in the upper layers, demonstrated that keratinocytes expressing the DRP1 mutant exhibit a spaghetti-like mess of elongated mitochondria, kind of like the extension cords on my desk right now. And histology of these cultures then showed us that failure of mitochondrial fragmentation impaired the morphogenesis of the tissue, which lacked typical features like basophilic granules, and they also retained nuclei in the cornified layers, shown as in the right image as a yellow arrow. So I hope I've shown you that organotypic epithelial models provide a powerful improvement over traditional in vitro cell biology. So our simplified model requires only fibroblasts and primary keratinocytes, which are relatively easy to grow. It replicates human tissue architecture, which exhibits some important differences from mice. Phenotypic data can be obtained in a couple weeks, and models can even be scaled up for drug screening. Moreover, the cells can be rapidly modified using viral or CRISPR approaches to target candidate genes. Finally, the cultures are amenable to live imaging, including the ability to treat the culture with vital dyes or cell-permeable labels like SNAP-TAG, or to transduce them with fluorophore-labeled proteins or optogenetic tools to examine real-time physiologic signals. On the other hand, there are limits. The cultures have a limited lifespan, but this can be extended by grafting them onto nude mice. And though the model I'm using does not generate epidermal appendages, this is an active area of investigation. So while the simple nature of the two-cell system increases experimental feasibility, there are many cell types not included in this model, like immune cells. However, co-culturing is possible of additional cell types. This has been done. And 3D printing may even make it possible to include pattern vasculature in the underlying supportive tissues. And so we're now aiming to move our science toward 4D cellular physiology with a focus on using advanced imaging to understand epithelial development and how it goes awry in disease. So there are many remaining questions that Janelia may be particularly suited to help address. Which microscopy methods are optimal for imaging stratified epithelia, including those that are thick and cornified? Can we perform single organelle or even single molecule imaging at native tissue interfaces in organoids or even intravitally? And how can we adapt our microscopy approaches for much longer term imaging at such high resolution? In a dream experiment, could we develop novel vital dyes and specific fluorescent tags to map all organelle compartments during the entire course of epithelial morphogenesis? So while my studies focus on mitochondria, we know the importance of interorganelle contacts to cell physiology. So how can we utilize distinct fluoropores to simultaneously image multiple compartments in cells chock full of organelles and cytoskeletal structures? I'd propose that a comprehensive understanding of organelle dynamics could reshape our approach to treat disease. So can we translate known human mutations in specific components into tissue models using gene editing to understand how exactly do they drive pathophysiology? And if we can use models to replicate human pathology in vitro, we can then use 4D cellular physiology to understand how to restore tissue homeostasis, how to slow the aging process, and perhaps how to mitigate carcinogenesis, which largely comes from epithelial tissues. And so finally, how can optogenetics enhance our understanding of organelle function by revealing their impact on dynamic fluxes of signaling mediators like calcium or reactive oxygen species? So using the advent of GCamp, we can monitor fluxes in calcium within live stratified epidermis. As shown in this video, calcium rapidly increases in the cytosol in response to the ER calcium pump inhibitor thapsigargan. Using tools like these may provide insight into treatments for genetic skin disorders affecting this calcium pump, like Derrier disease shown here from one of my patients. So other optogenetic tools like killer red and photoactivatable GFP can also be targeted to specific organelle compartments to allow spatiotemporal control of organelle labeling and damage while permitting us to track their fate in live cells. And we can now move these tools into epithelial models to understand how intact tissues cope with injury to organelles, how their inheritance affects daughter cell fate, and whether organelle repair mechanisms can be identified and then augmented to promote resilience to damage or aging. So with that, I'll give a huge thanks to my mentor, Erica Holzbauer's Penn, as well as Andy Moore and Marco Toquito for helping guide advice on this work. We collaborated with the University of Michigan for CRISPR experiments, as well. I actually had the chance to visit the Advanced Imaging Center at Janelia a few years ago, which really inspired me to think about novel imaging mechanisms that could advance our ability to understand the epidermis. And finally, I'm grateful for the various sources of funding that have supported my work and my career development. I thank you all for your attention. I look forward to our discussion.
Thank you, Corey, for that really nice talk. I actually want to take a moment and let everyone know that Elaine has not been able to join us yet. She's having some internet issues, but she should be back on shortly. So I think what we will do is move straight into the discussion, and perhaps we can entertain some of the questions that have come in for Rick and Corey in the meantime. And thank you both for great talks. And let's move on to the discussion.
Fantastic. Yeah, thank you all so much. Really exciting. I think that the two approaches that Corey, you and Rick articulated are a perfect merger in a sense. Doing human genetics to identify pathways that are impacted, and then coming in with these in vitro organotypic systems to actually interrogate how a particular gene mutation is functioning is a super powerful combination of approaches. I see that Michael has a question, so why don't we just jump into your question, Michael?
Thanks. So great talks, both of you. Rick, my question is for you and relates to maybe complex phenotypes that are associated with monogenetic diseases, but ones that produce cell autonomous, but not necessarily gene-specific phenotypes. So for example, Corneille-de Lange syndrome, where mutation of a single gene leading to the disruption of a single protein produces a phenotype that's due apparently to relatively small changes in the expression of a huge number of individual RNAs. And I'm wondering, to what extent do you think that genes like that, with complex phenotypes associated with many, many genes, might complicate this sort of genetic analysis that you're talking about?
Yeah, I think as we get farther into studying mutations that cause developmental abnormalities in humans, we're seeing this quite frequently. So for example, we've recently completed a study of about 12,000 patients with severe congenital heart disease. The most frequently mutated genes in these patients are chromatin modifiers, specifically H3K4 methyl readers, writers, and erasers. But others as well. So these are exactly as you would suspect. So they affect the expression of many different genes. The phenotypes are quite diverse. And get back to my earlier point of there's considerable variable expressivity of the specific phenotype, which again suggests the possibility that there are critical points in development where you need the right dose of the right gene at the right time in order to get down the right path for development to proceed in that tissue properly. And if you don't, you end up with a phenotype in that tissue, but it doesn't mean that you're going to get all of the different tissues that could be affected in any single patient. And in fact, one of the most striking observations in this analysis and the autism project that we've had a more tangential role in is that there's striking overlap in the specific genes that are mutated in both autism and congenital heart disease, which would not have been commonly expected between the intersection of these traits. Suggesting that, again, there's variable expressivity. So it doesn't mean that you can't get discrete phenotypes from mutations that have protein effects on gene expression, but you may find a lot more variability than you bargained for.
Wonderful. Elaine, are you back to, can we listen to you now? Yeah.
Who knows? Let me just see. You can keep asking questions until I'll go ahead and try to set up my presentation. Okay. Sorry, the computer, the internet in my office crashed, so we'll try it again. Can you see this?
Yes, we can see you. Yeah, this looks good.
You can see my presentation?
Not yet.
Not yet.
Just share screen.
Okay, let's give it a try. This is crazy. It could be that my Zoom might have crashed with.
Could you remove your double screen? Just have one single screen.
Let's give it a try.
That helps a lot.
Yeah, let's give this a try. See, I don't have. Let's see. I don't have an option of sharing my screen on this thing. Let's see. Maybe. Okay, let's give this a try.
That's good.
Can you see that?
No.
Can you double click? Yeah, this is good.
Yeah.
Perfect.
See it?
Excellent. Yeah.
Okay, we'll give this a try. It could be that the whole system is crashed, and I hope I don't have to reboot this. Anyway, I really apologize.
This is great. Perfect.
I'll call my president immediately thereafter the session. We'll try to figure this out. Okay, so I want to start just by dealing with some broad issues, and this is really considering the world's major health problems. And as I was thinking about what to talk about, we really have escalating climate change. We have contaminated foodstuffs and water. We have air pollution, allergens. And we're facing increasing risks of cancer, of chronic inflammation and allergies, and of an inability to cope with ever increasing emergence of new pathogens and microbes. And that's really evidenced not only by our current pandemic, but on Ebola, antibiotic resistant staph infections, and so on and so forth. And I think bearing the brunt of all these diseases and disorders are epithelial tissues. Those are the lung, the intestine, the skin. They're really forming the body's barrier to the external environment. And we work on the skin and the epithelial, but the epithelia of our body basically can constitute our primary environmental interfaces. And in the skin, as in many of the other epithelial tissues, we're dealing with mechanical stress, noxious agents, metabolic stress, pathogens, wounds, temperature and pH extremes may be more specific for the skin, commensal microbes, oncogenic stress, and then ultraviolet light, again, more specific for the skin. But all of the epithelial tissues of our body have stem cells, and these stem cells have to be very active and able to be able to sense all of these different changes and to be able to rapidly adjust. And that's what my laboratory is interested in. And dealing with epithelial tissue stem cells, we know that they are responsible for making, rejuvenating, and repairing the barriers to the outside world of pathogens, microbes, allergens, and environmental extremes. And we also know that they reside in protective but dynamic niches, which through a myriad of complex and still poorly understood communication networks control stem cell behavior. We heard this morning that there's 58 different cell types in the lung. There are 65 different cell types in the skin. So that's one of the reasons why, despite working for 10 years on organoids, when I first started my laboratory, we've shifted to looking at an in vivo system. And I think with the increasing contributions of people like Jim Wells and others with regards to organoids, that we can start to go back and forth between using mouse genetics and human genetics to exploit some of the things that we cannot do with regards to organoids, such as looking at some of these really complex communication networks. So epithelial stem cells reside in tissues for the long haul. That's another important consideration. And if they malfunction and don't form the barrier properly, or fail to communicate with the immune system correctly to grapple with infection or wound repair, then the disease risk escalates. And so I'll just briefly touch upon some of the things we've been studying. Atopic dermatitis. This is a very common inflammatory skin disorder, one that's often thought to be an immune disorder. The treatments are immune suppressive drugs, but consider the fact that there are environmental contributors, cold and dry environments, and also consider genetic determinants, such as mutations in genes encoding filaggrin and filaggrin paralogs. These are not immune cell genes. These are epidermal terminal differentiation specific genes that are produced by the stem cells as they produce all of these outer layers that form our skin's barrier. And for a long time, it hasn't been clear, because this is a very large protein, whether or not this has anything to do with the disease with regards to these mutations. They reside throughout the gene. The gene is encoding a protein that contains repeats. These repeats are rich in histidine. And so this has been a mystery for many years. And we got into this mystery by virtue of having a biomedical engineer join my lab. Again, one of the important aspects of studying epithelial biology, you really have to go beyond simply the epithelium if we're really going to understand epithelial biology. And it turns out, to make a long story short, that filaggrin undergoes thermal, pH, and concentration dependent liquid phase transitions, first forming membraneless granules in the terminal differentiating cells, and then dissolving them in the transition from the living layers of the skin to those dead flattened squames that are providing the protective barrier of our body surface. And patients with these mutations, as we've generated them, we've shown that they cannot undergo these liquid phase transitions. They're defective in that process. And the consequence of that, both in mice and in humans, is nuclear retention and skin barrier defects. And that is what increases the risk of inflammation in these various different patients. And so, again, we really need to understand the basic nuts and bolts of epithelial biology in order to be able to start to appreciate some of these very complex and seemingly surprising aspects. I'll deal with another, really going broadly to chronic inflammatory disorders. These are common, not only atopic dermatitis, but psoriasis, chronic wound healing, asthma, inflammatory bowel disease, which you've heard about to some extent today. The hyperproliferation comes and goes, but with each subsequent assault, it often recurs in the same spots quicker and more robustly. Again, a phenomenon that we really haven't understood very much until several years ago, when we started to look at the epigenetics that were going on in response to, as the stem cells are responding to these various different inflammatory stimuli. And what we realized is that there is a retention by these stem cells of epigenetic memory. It's buried in their chromatin memory of their inflammatory experiences. It's a mark that basically gets activated the next time an inflammatory assault arises. And the immune system participates in epithelial memory, but it's not required to maintain it. So it's really maintained by the stem cells themselves. The memory gives the stem cells the ability to activate in a much more fast and robust fashion to subsequent and not necessarily the same inflammatory assaults. And so when you start to think about it, ask the questions then of, do stem cells harbor memory of different experiences? Is memory cumulative? If that's true, what about aging? And what about various different inflammatory assaults? How does memory contribute to cancer? And when we think about it, as we've been learning over the years, there's an immune stem cell complex array of interactions. They're a two-way street. The immune cells signal to the stem cells cells signal to the immune cells in a whole variety of different orchestrated events that allow our stem cells and our epithelial cells to be able to do the right thing at the right time. And when we think about disorders like, think about the most recent pandemic, for instance. Here we have a situation where it's aging individuals that have a tendency to suffer the most severely and produce these cytokine storms. This kind of phenomenon could start to explain the various different phenotypes that we see in response to robust new inflammatory encounters. And so I think it's important to really dig into the mechanisms. Also, when you think about that at the mechanistic level, if we could simply not treat with immunosuppressive drugs to treat these patients, which really doesn't take care of the problem, but rather focus on perhaps erasing inflammatory memory, we might have new types of treatments for these diseases. And then I'll just finish with the notion about cancer, because again, many of the most deadly and most common cancers derive from epithelia. I've listed a few of them here, but there's a very long list of epithelial-based cancers. Tumor-initiating cells, the so-called cancer stem cells, typically originate from oncogenic mutations in long-lived stem cells. And so again, they're equipped by the time they get to cancer with an arsenal that enables them to evade chemotherapy and immunotherapy, and they're not necessarily the highly proliferative cells of the tumor. And so we often, with various different tools to be able to treat cancers, don't get the cells that actually matter and are responsible for regrowing the cancer. And so to develop effective therapeutics that really selectively target the cancer, we need to be able to target the right cells, the cancer propagating cells, and we also have to know enough about normal epithelial and stem cell biology so that we can target against the cancer stem cells and not affect the normal stem cells, because that's what makes people sick when we hit the wrong cells and start to hit natural cells. And so again, these are areas that my laboratory has been very interested in. We've been isolating and characterizing where the stem cells or the tumor initiating cells are. We've come up with genetic mechanisms to be able to mark and monitor and track. And by looking in vivo with high imaging characteristics, we can see that wherever blood vessels come near these stem cells at the interface, that changes their properties. And because we can do genetic lineage tracing and those kinds of things working with mouse genetics, we can mark and trace these cells. We can show that these are the cells that when they receive a TGF-beta signal from the perivasculature are the ones that are breaking down the basement membrane and evading. And we can also show that in response to various different either chemotherapies or immunotherapies, as we've been working on, that they are the cells that survive these therapies, and they are the cells that are responsible for tumor relapse. And so for the sake of human survival, Janelia needs to make epithelial biology a priority.
Thank you, Elaine. That was a really nice talk, and I'm glad that we all got to hear it. So I'm going to go ahead now and turn it back over to Jennifer and Jitu to continue the panel discussion.
Great.
Wonderful.
Terrific. So Jennifer, I think there's an interesting question for Rick in the question answers. So perhaps if Rick would look at that question, and I'll probably rephrase it a little bit. In terms of the questions about, can you uncover or determine new strategies and technologies to uncover vulnerabilities in genes, which could influence, physiology of the whole animal. And I guess, are, for example, signaling networks, the sites where such vulnerabilities may exist. So that is a question,
Yeah, I think the biggest challenge with genetics is also its biggest advantage. You really don't know what you're going to find when you start with an interesting phenotype. You frequently don't even know what organ. As the hypertension work highlighted that what organ is going to underlie this trait. I think the art form in genetics is picking interesting phenotypes, and clinically severe phenotypes that have not been explored turn out to be remarkably rich, and there's still plenty of them out there. In particular, in development, all of the morphologic abnormalities that are typically not seen in a lot of academic medicine, they're seen in orthopedic surgery and other areas that don't have such a great clinical investigation, are vastly underexplored. And then there are other phenotypes that I also think are underexplored that are, surgical complications, acute kidney injury that Michael knows a lot about is a good example of a trait that may not have been under strong evolutionary pressure because whoever had surgery before, 100 years ago to experience acute kidney injury after coronary bypass or other major procedures. So I think there are a lot of phenotypes that haven't been explored. You just can't pick which axis you're likely to find things in, with a few exceptions. Craniosynostosis, these have turned out to all lie in genes involved in regulation of BMP, Wnt signaling, and RAS signaling, for example. But I think there's an awful lot that remains to be discovered.
So we have some participants who that are on the screen with us. I want to start with Dennis and then Kathy Green, if you guys could share with us your questions. Dennis?
Yeah, Elaine, that's a great talk. It's a question I've been asking some of our clinicians at MGH is whether it's worthwhile having the second COVID vaccination in a different arm to avoid a local enhanced inflammatory response based on what you've just shown us.
Yeah, certainly in terms of especially a month apart, we looked at up to 6 months, and we still see epigenetic marks of inflammatory response. And again, it doesn't appear to be systemically as severe. So we've looked at the other side, and the other side doesn't get affected in the way of the site of inflammation. So we haven't, let me preface this by saying we haven't looked at COVID, but I think it's really interesting. I should also mention that since we did these studies, people have also now demonstrated epigenetic memory in lung epithelia, and you think about asthma and all these other things. And they've also demonstrated it in hematopoietic stem cells. And in hematopoietic stem cells, in contrast to what we've seen, but I think as interesting, that in hematopoietic stem cells, sepsis will actually cause skewing. It'll leave a mark on the hematopoietic stem cells and actually cause a skewing toward the myeloid lineage, which basically is capable then of fueling an inflammatory response. So it could go beyond simply that of since COVID affects so many different epithelial tissues and so many different tissues, it could go beyond that as well.
All right, thanks.
Kathy?
Thanks, Jennifer. Yeah, so I had sort of two areas to discuss. The first of which, going back to Corey's really beautiful talk, you gave a really good example of how one might use these 3D organotypic epidermal morphogenesis systems to address the question, but you also talked about some limitations with regard to the presence of other cell types. And then Elaine told us there's 65 different cell types in the skin. So I'm wondering, and she also mentioned going back and forth, that these two kinds of things could inform each other. And Corey, I'm wondering, if you've thought much about to what extent we can develop these ex vivo or organotypic types of systems, bringing in neurons, immune cells, possibly using 3D printing, not only for vasculature, but maybe other cell types. I thought I'd throw that out. And then I had a question for Elaine about mechanics and epigenetics, because you showed pictures of psoriasis and atopic dermatitis where, you know, you see these lesions occur in the same places, like on the outside of the elbows, the inside. How does that contribute? So two very different questions. Corey, maybe you could start with the first one.
Sure. Yeah, so I think it almost gets a bit overwhelming if you try to think about, you almost have to appeal to some sort of augmented intelligence to make sense of all the different interactions that are happening in the skin. It is a repository for long-lived memory T cells and B cells even that stay there and are almost like a lymphoid-associated tissue in the skin. And so I think you can't ignore those things. I tend to be the type of biologist who likes to understand the simple problem first before we sort of expand into massive amounts of data that we don't even know how to organize or make sense of. So I've sort of focused on what's the epithelial compartment getting towards the barrier effect of the skin, which becomes compromised in a lot of diseases. But absolutely, you could think about infusing immune cells into the model, like you said, imprinting lymphatic channels and vascular channels, and actually having more of a microfluidic kind of environment where there is exchange of nutrients and metabolites that would more closely reflect human physiology. There's been at Northwestern where they were able to make the sort of in silico ovulatory cycle, basically, like they could replicate the entire physiologic cycle of an ovary in a small dish. I mean, it's amazing what you can do now when combining engineering with epithelial biology. So I think it's a great place for Janelia to go as far as harnessing some of these other technologies and bringing them to epithelia, which do create a huge burden as far as disease and cancer. So that's a great thought.
Great, thanks.
Yeah, with regards to your question, Kathy, thanks for raising that. One of the things that we see is in epidermal differentiation defects, flexural regions are the areas which are most affected because they are the regions where your skin barrier is really challenged to a height. And so when you look at some of these diseases, it's not to say that there aren't contributing factors for the immune cells. There certainly are. If the immune system goes haywire, that can exacerbate some of these situations, but it's surprising how many and how many of the different types of always thought to be strictly immune disorders and all treated with immune suppressive drugs are actually disorders that involve mutations in that cause an alteration in epidermal differentiation. And so I think the two are there together. Effectively, if the barrier is breached, then that's going to trigger an inflammatory response. If the immune system is haywire, that's going to trigger. Could trigger also barrier defects. And so I think these are really important issues. That said, the whole issue of mechanics and epithelial biology, I think, is a whole nother very interesting area.
So as far as tension versus compression in those areas, do you think that those are also talking to the nucleus and through the cytoskeleton to inform the epigenetics in those places?
It could well. We've recently measured using atomic force microscopy and various other tools to measure where the mechanical forces are coming from inside the skin itself. And what we found is that the stiffest areas of the skin are in two places, both of which are produced by epidermal stem cells. The first is the outermost layer of the skin, which is much stiffer than any of the other epithelial cells or the dermis. And the next is the basement membrane, which is much stiffer than that of the extracellular matrix within the dermis. And so it's really that the stem cells are architects of their own mechanical forces, and yet those mechanical forces can really wreak havoc when you couple that with genetics and genetic mutations that are going to perturb overall tissue structure. And that in turn is going to be impacted by alterations in mechanical forces. So
I was going to make one other comment about the mechanical force thing, is that if you go from clinical morphology, it really informs you about the biology. So lupus, for example, tends to happen on the knuckles, which gets stretched a lot, whereas its counterpart, which is a seemingly similar disease, dermatomyositis, tends to go between the knuckles. So there's something about the simple observation and the like classic physical exam that can perhaps inform the way we think about what's driving these immunologic changes.
And areas between the knuckles are basically, or areas between the fingers are going to be areas where humidity is going to play a role. And humidity and temperature also play a role in not only the liquid phase transitions and filaggrin, but we've also demonstrated that the keratin tails of the terminal differentiation specific proteins are also unstructured domains that are sensitive to liquid phase transitions. And so it looks like the whole terminal differentiation process of the epidermis has been that the stem cells have exploited over evolution the extremes that we're facing of temperature, of humidity. Also, our body surface is pH 5.5, and it's exploiting the environment to take advantage of that with regards to producing an optimal barrier. So I think these areas are really fascinating about how we can't really just be epithelial biologists. I was so pleased to hear Anne's talk today and some of the other talks today where people are bringing in these various different areas. And I noticed there's a workshop on regeneration and wounding that is headed by one of my former postdocs. And you know, it's really all, I think, a real focus. I'm not sure you can really identify one single area at Janelia to go for, but I would say we need, at the center of all of that is, even at the center of the nervous system is the epithelium, because basically my interest in the nervous system is that essentially it's progeny of the epithelial cells.
So with that, we've got three people with us that have their hands raised. I'll just go from right to left on my screen. Maha, and then Michael, and then Props. So Maha, you want to raise your question?
Sure. Thank you for a very nice talk, Elaine. And I was going to ask you which system of, or which aspect of epithelial biology is best understood in the context of epigenetic memory. By that, I'm going to be specific and ask, the type of an environmental change, the magnitude and the frequency, because any one of these presumably can lead to either short or long-term memory, and that can be in time and can also be in space. And I'm just curious whether there is one system which is reasonably well understood where one can start asking exactly what you were raising, more quantitative perspective.
Yeah, I mean, a good portion of my lab right now is working on this problem, but since we only published it for the first time in 2017, we don't know that much about the answers to your questions. But what we are learning is that the memory seems to be harbored in genes that are very robustly responsive in the first inflammatory assault, whatever that may be. And so it's kind of suggestive that whatever gene needs to respond to a particular environmental condition and to really jack up their level of expression, that those are going to be genes that need to have memory. And the other aspect that we're learning is that our first glimpses, while we still have much more work to do, but our first glimpses are that memory is cumulative. And we showed in the first instance that it can last for 6 months, but it can then still be accumulated. So
may I just continue that and ask, when you say if it's cumulative, are you saying that if I have a small environmental insult for a long period of time, it's exactly analogous to a large one for a short period of time? And
no, I don't think so. I think that although we don't know that much about it, but we do know that the ones that last for a long period of time are ones that are robustly activated at the first inflammatory assault. We do know that the second inflammatory assault need not be the initial stimulus, so that if you give psoriatic-like response in the first case, you end up conferring a more rapid response to yeast infection in the second case. And that's something that has been known in the immune system as well. Innate immunity or innate epigenetic memory is basically similar. The second is salt. These are general at the epigenetic level. They are general. They're not like B and T cells, which involve T cell rearrangements or B cell rearrangements. So that's about all we know so far. But we're currently testing long term. The problem.
And is there crosstalk? You mentioned temperature, humidity, mechanics.
Well, those kinds of issues we haven't even touched on. But we have. I do think that you ask why would the human body want to do this if all that we're getting as we age to be just a mess of various different inflammatory reactions, and generally they're deleterious. So why would we have evolution that keeps that? I think it's because of wound repair. Because in a wound, you have to repair the wound as fast as you can, and so I think evolutionarily that's why we have these kinds of mechanisms in place. It's just that we weren't designed to live as long as we do. And as we accumulate these memories, that's where really chronic inflammatory diseases are something that are really age related. So, and the current understanding is not really that well understood. It's not understood at any level yet about these things. Even in the hematopoietic system or the macrophages, it was first described for 6 days. The macrophage has a short half-life, and that was called innate immunity or epigenetic memory. And now that there's hematopoietic stem cells, well, they're long-lived. And so now we're beginning to get understandings, but all of this is it's a new field.
Well, let's move on to Michael Caplan with your question.
Thanks. So I have a question for Corey. We've heard a lot so far today about the role of mitochondria and metabolism in affecting function of a variety of cells, including epithelial cells. You showed very nicely in your system that mitochondrial dynamics seem to be incredibly intertwined with epithelial differentiation. And you actually did something that I think is a really nice new experimental paradigm, which I would call interventional cell biology. And that's something I'll talk about a little bit more in my talk. The idea of being able to not only look correlatively at changes in organelle structure, but induce those changes and see what the consequences are. I guess what I'm wondering is, do you have any idea how that change in mitochondrial morphology affects mitochondrial function, and whether it is a change specifically in mitochondrial function that induces differentiation? If you coat the skin with a mitochondrial uncoupler, for example, do you produce similar effects?
Great. Great questions. I mean, I think the role of mitochondria in the aging process, in disease processes, in the immune system, transfer of mitochondria from one cell to another, like this stuff is happening in the body and can really be a very cool window into pathophysiology. So I think probably during epithelial differentiation, there's a programmed manner in which organelles are broken down so as not to do these things prematurely, because that would be deleterious. If you thinned your skin into a single layer, you wouldn't survive very long as a human. So you have to have these things programmed in a way that you're allowed to have a sort of certain thickness of epithelium. So I think the mitochondrial respiration, their generation of reactive oxygen species, but they're also a minor storage compartment for calcium, which is a major signaler in the epidermis. We've known that for many years. So maybe keeping mitochondria intact until a certain point in differentiation may be essential to keeping keratinocytes into a pre-cornified state until they're really ready to kind of flip that switch and go through the phase transitions and everything that Dr. Fuchs was talking about. Once that stuff happens, I don't think there's any going back from there. So I think there's got to be a huge regulatory network making sure the mitochondria don't release things too prematurely. it's not really typical apoptosis that happens in the epidermis. That happens, but in response to like toxic stimuli. It's a different form of cell death that has to be very regulated to make sure that the epithelium stays a certain thickness, and that ranges over different parts of the body.
Great. Props, do you have a question?
Yeah, actually, I had a question for Corey, kind of related question to what Michael asked. So basically, your data, you showed that the change in the mitochondrial morphology is one of the big factors for differentiation. But I was wondering, like, you do have also a gradient of nutrients and oxygen, right, along the axis as the cells are differentiating. Now, how is that coupled to the differentiation process? And also, I was thinking, are these cells kind of isolated, or are there basically gap junction mediated signaling going on, which also plays a role in this differentiation along the axis?
Right. Regards to gap junctions and coupling, I think junctions are pretty well maintained in this type of a model, but we now have optogenetic tools where you could directly test that. You could have a channel that can be activated or deactivated and watch fluorescent molecules transfer. So that could be tested now with the fluorescence tools that are out there. As far as gradients, I mean, this system was set up, by Dr. Fuchs and Green and everyone who understood that there's this interplay between the fibroblast feeder layer and the keratinocytes. That's absolutely essential. So there has to be a gradient if you only have fibroblasts on one side of the epithelium. There has to be a gradient if you only have blood vessels on one side of the epithelium. So these cells are sensing some sort of real live gradient as we're all sitting here. One side's exposed to the air, the other side's exposed to much more of a nutrient-rich milieu. So I think that's got to be part of the signaling process that tells these cells at some level they're either relatively starved or hypoxic, and that probably triggers a change in their metabolism. And a lot of that's being worked on with metabolomics and things right now. And that's not my area of expertise, but I think there's definitely a gradient there.
And mechanics as well, Corey.
So Jitu, do you have some things to add? Did you want to add some things?
Yeah, I was just wondering if one could think about how one might look at the genes involved in setting up memory and erasure in the systems that you were looking at, Elaine, and whether there is any evidence for that already in sort of genetic diseases that Rick has been talking about. I mean, I'm just wondering whether one could think in those terms, whether the physiology could tell us something about these mechanisms from their perturbations.
Yeah, I mean, we've certainly, one of the interesting aspects of that is that if you look at memory, first of all, we're actively working on the molecular mechanisms of memory. We and everyone else in the field who works on memory. And we have a pretty good understanding, which I won't go into today, but I think that's an important area, what controls memory. But the other aspect that you're really raising, G2, is what about the genes that memory activates? And one of the very curious aspects is that whether you're a hematopoietic stem cell or an epithelial stem cell, that the response there's a huge overlap in terms of the initial response, what genes are getting turned on. And there is some insights into that, because what you do find are various different metabolic genes, for instance, which would be straight at the heart of the kind of question that you're asking, G2, and the kind of thing that Corey was talking about. And so, and then there are other classical inflammatory, how do you set off the communication network, for instance, initially, because the epithelium is the one that really is acting as a baton in this. It's the one that's sensing the signals from, say, a bacterial infection or a pathogen infection, and it's relaying that to the immune system. And so its response, some of these initial responses are really reflective of these kinds of relay systems. And if you're in the hematopoietic stem cell, you really rely upon hematopoietic stem cell progeny for the various different recruitments of what do you actually need to fight off an infection. And those are actually quite common. And people look at them. They're still pretty poorly understood on all planes, but the mechanisms of the initial, what do we need to recruit to fight off an infection? There's a lot of parallels across not just epithelia and hematopoietic systems, but muscle, many different systems. And so I think it's going to be an emerging field. And the more we know about it, obviously, the more insights we'll have to this.
Well, thank you so much, Elaine. And we have to bring this session to a close. I want to thank Rick, Elaine, and Corey for really spectacular presentations and discussion. We are now going to move into our fourth session, which is tissue self-organization. And I think this is a really interesting sort of transition from the sort of genetic, sort of fundamental cell biological approaches that involve protein interactions to one where we're going to be thinking in a different way in terms of self-organization. So our panelists will be Zev Gartner, who's from the University of California, San Francisco, Thomas Lequay from Marseille University, and Colby Chinaweski from Vanderbilt University. So Zev, or I should also say that the discussion is going to be led by Patrick Chitwood, our Janelian postdoc, and David Klapman, who is a vice president, HHMI, and has a lab at Janelia. So Zev, please take it away. Thank you so much, Zev, for that really wonderful talk. We'll now move to Thomas Lequay.
Yes. Hello. Can you see my screen?
Yes.
Okay, so thank you. It's a real pleasure to be late at night and like having dinner with you, exchanging ideas in a very open format. So I'd like to entertain you with so my fascination for many years about the problem of morphologies, and really in the context of this workshop and this session, I want to indeed talk about self-organization. The one thing which I think is the most striking feature is the fact that phenotypes exhibit a very remarkable stability. I mean, several of us have already mentioned that. And I mean by that there are invariant features that are there despite internal renewal of all the components in tissues. And I mean by that cells, obviously. And this is quite a remarkable property that is found in organs, in embryos, and all of this. And this has really been a fascination for us to try to understand what underlies the stability of phenotype. And what I would like just to throw in first as an important aspect of this in answering this question is that of information, which I'll come back to later on, which in a way allows a way of encoding shape and size in embryos and in organs. And so in thinking about this information, I'll come back to that in a second. I would also like to say that on top of this, what allows the stability of phenotype is the fact that there is internal dynamics in every single organ or embryos that is made out of epithelial tissues or even mesenchymal tissues. And this dynamics indeed allows for some restoration of the macroscopic phenotype in the face of cell death, cell division, cell extrusion, all of these seemingly random events that happen in any tissue as it grows and acquires its shape. And so dynamics is not actually something which prevents stability, but rather the condition for the restoration of phenotype stability at any time in the life of an organism. And the last thing which I would like to say here is that on top of all the internal information that is required for the production of shape, there's also, of course, a function which may provide a mechanism for feedback that actually ensures that the shape of an organ, whether the size of a cavity or other structures, are actually maintained over time. So in thinking more specifically about information, here we have 3 movies that display at the cellular scale few cells. So we have a green alga on the top left, then we have a sea urchin embryo undergoing cell division in the early steps of development, and later on we have a zebrafish embryo. There we can clearly see that what information is really doing is providing a way of encoding internally space and time. And this is really something which, of course, we have a sense is encoded genetically, but I would like to slightly broaden this perspective in the context of this session in particular. So on the top left, we indeed have genetics and biochemistry, which are sort of the core pillars whereby information is encoding the size and shape of epithelial tissues or any tissue. And by that, I mean really sort of the patterning information that breaks symmetry, defines axes at the tissue scale. And at the cellular scale, we're talking about polarity, whether it be the epicobasal polarity of an epithelial cells or its planar polarity. And these are essential sort of input to the information. But while this is a traditional way of thinking about information in epithelial tissues, I would like to add to that what is not just an output of genetics and biochemistry, but really sort of an internal component of morphogenetic information. I mean by that mechanics. Why? Because mechanics basically provides a means to define length scales and time scales to really pattern a tissue, when we have several examples of that in mind. By mechanics, I mean a number of things, and sort of the most obvious things in the context of epithelia would be adhesion, sort of the cohesive forces between cells, contractility, which in the form of actomizing networks provide active processes that enable plasticity at cell boundaries within cells to really deform a tissue, and also hydraulics, which is the outcome of the outcome of the, and indeed what a flux is allow for the emergence of pressure, which is one of the active elements of cell and tissue mechanics. And the last is also equally important, is what so it's gone by the broad term geometry. I mean by that any tissue will have a characteristic dimension, which could be quite diverse, curvatures, and more generally would define sort of the boundary conditions for mechanics and biochemistry to play their role during the development of a structure, in particular an epithelial organ. So really these 3 modules of information interact with one another and define sort of the core pillars of morphogenetic information. And so the question that I would like to bring in then is really sort of the modes of information flow within this network. And the first one, sort of the traditional way of thinking about information flow, is one that actually has been already used by several of us, which is the term program, which I use here to refer to the fact that information flow could be very hierarchical, flowing downward in an invariant way, the rules of interactions being deterministic. And this is typically what is associated with the notion of gene activity controlling many of the cellular and tissue processes. Associated with that is also the notion of modularity. And indeed, there's a certain way of understanding, for instance, how the making of an embryo, where fly embryos or zebrafish or other organisms, really follows from genetic patterning, which really controls directly at the cellular level cortical mechanics, cellular behaviors, whether cell movement, cell division, and so on and so forth, and how you can really predict tissue morphogenesis from the information that is laid out at the onset. Now, it turns out that there is a more, I think, equally useful, if not more useful, way to think about the emergence of pattern, which is self-organization, which I strictly refer to by saying here that there's no hierarchy at all. There's no pre-pattern. We start from a homogeneous medium of cells, which are all alike, and through local interactions, whether mechanical, biochemical, mechanochemical more generally, and the existence of stochastic fluctuations and feedbacks, some emergent pattern occur. And in this context, the information relies on sort of the interactions among biochemical cues, mechanical cues, and geometry. And we have a number of examples of such processes. I just cite two on the right, which you're all familiar with. One, obviously, is the emergence of vilification in the gut of different organisms. And the bottom is actually the development of the branch pattern in the mammary gland, for instance, which is the work of Hannes and colleagues. But there are many other examples of self-organization building upon the principles that I just mentioned on the top left of this slide. So having this in mind, I would like to then move on to what I identified as very general and important in this context to understanding organ shape and organ size. So the first is that having that in mind, we can see how genetics in particular on the left and the environment on the right provide means of control of this flow of information within cells and tissues. And in particular, the environment provides a means to actually tune whether the mechanics or biochemical activity, and thereby to actually control the system in a particular way. And so three general questions that I identify would be first, how regular shapes emerge from the stochastic processes occurring at the cellular level. The second important problem is that of the recursive nature of information depicted as I've shown here. That way of recursivity among biochemistry, mechanics, and geometry provide the means for an adaptation to whether internal or external modes of perturbation, such as the environment or genetic mutations, genetic lesions, but as well as, of course, the intrinsic regulation of the systems during development and in the adult. And the third general question is the problem of coupling shape and size in such organs. And really the questions which I would like to finish with, which is the question of how size is encoded in such context. So imaging epithelial tissues such as this one so this is here the imaging of a Drosophila wing imaginal disc. We can see over many hours many cellular cell division events, which are zoomed on the right. And these tissues keep their barrier function while they divide. And the remarkable features of any organ, such as the wing here, is that it grows exponentially, and then it attains a target size. It transits from an exponential mode of growth to a linear mode of growth, and then arrives at a steady state. And knowing what an exponential growth is, we realize that there has to be exquisite control over the arrest and the slowing down of growth during development. The second point is that there are feedbacks to maintain the proportions between paired organs and within the organism. This point actually was already mentioned by Helen before. This is truly a remarkable features in the wing. The error is within 2% in this system. And at steady state, the growth persists in other tissues, such as the gut, and allows thereby the regeneration and the adaptation to the environment. So having in mind what I presented before, biochemistry, mechanics, and geometry as the three pillars of morphogenetic information, we indeed can explain a number of things that explain growth arrest. And there have been numerous models of morphogenetic gradients, mechanical feedback, and other things that provide some layers of control for the arrest and of growth in this tissue, which I'll not go into. But what I would like to emphasize that these studies have largely ignored an important element which I would like to focus on now until the end, which is the energetics of the process. So by the energetics, I really mean a number of things. The fact that energetic is associated with, there's a certain demand of energy for the system to grow, and obviously for the metabolic reactions, anabolism, catabolism, but also for the signaling events that are required for actually even the very control of size. Both are energy consuming processes. The second is that there are internal constraints associated with the energetics. The first is is an external constraint, sort of the supply of metabolites and the oxygens to all the many cells of an organism, 310 to the power of 13 in humans. And the internal constraints are also important, such as the constraints imposed by the synthesis rates of mRNAs and proteins, for instance. The third point is that there is regulation and adaptation of this system. There are many internal embedded feedbacks at the organ scales as well as the organism scales, as well as a response to the environmental inputs, which could be nutrition, obviously, which do affect the way that energetics is regulated. And really, the specific questions, which I think are very open because we don't know much about it, are really the interactions between energetics and mechanics, energetics and the genetic programming of these systems, as well as even the geometry in the sense that I've defined before. So these red arrows really point to questions to be investigated. And I would like to just show in numbers the issues of metabolics at the cellular level within the context of the organisms.
Hey, Thomas.
So for a human being,
I'm sorry
yes,
to have
it's finished.
to cut you off.
Yes.
Are you on your last slide?
This is my last slide here.
Okay, thank you.
So the key questions are really the phenomenology which is missing. There's no measurements of the high precisions of cellular volume, mass, or growth dynamics at the cellular level. We need to come up with means to measure the microcalorimetry at the organ scales and even the cellular scales. We need live metabolic probes, this has been said. And we also need to have ways of controlling and perturbing by optogenetics the mechanics of cells to decipher the feedback control network across scales. We also need a way to have a sensitive system that will allow us to control the energy supply and monitor the response with high precisions at the cellular scale. And last, we need a way to model all of this information into a predictive theoretical framework. Thank you very much.
Thank you so much, Thomas. That was really wonderful. We'll now move to Colby Czernowiecki. Sorry if I mentioned your last name incorrectly, but please share
It's easier than what most people think it is. It's just Czernowiecki.
Czernowiecki. Okay, cool.
All right. We are full screen here. Everyone can hear me just fine?
Mm-hmm. Great.
Fantastic. So I just wanted to give a big thank you to the organizers for allowing me to present at this wonderful workshop. I think it's really great, all of the topics and areas for innovation that have been brought up today already. My name is Colby. I'm a fifth-year PhD candidate in Matthew Tiska's lab at Vanderbilt University. And today I'm just going to talk to you a little bit about a specific contractile protein, non-muscle myosin 2C, and its role in the intestinal epithelium. So personally, I think the small intestine is a fantastic example of complex and dynamic epithelia. So while the small intestine is inherently a tube, as you can see in this image on the right, it is of course covered in all of these finger-like protrusions called villi, which help allow for the additional surface area that's necessary for nutrient absorption. These villi all point inwardly to a central lumen. If we want to take a closer look at just one of these villi, using immunofluorescence microscopy, we can see that there's some really fascinating structural features in this tissue. The first one being that this is divided into a crypt domain and a villar domain. This crypt domain harbors an active stem cell niche. Those cells will differentiate, and then they must begin their journey to migrate out of the crypts, out onto the villus, where they will eventually be shed out at the tip. So the steady state for the intestine involves this process of cells constantly differentiating, migrating, and shedding. A cell can start at the base of the crypt and migrate out to the pit in anywhere from a few days to over the course of a week, depending on the organism you're looking at. And so because of this, understanding the process by which all of these cells develop and then migrate is really important, because that steady state constant turnover is necessary for the organ's function. Something else that I think is of personal interest is that within those when the cells exit the crypt and come out onto the villus, they also have to undergo a fairly dramatic shape change. So when cells are down in the crypt, they're going to be concave. They're going to have that inward curvature that we see in a lot of internal organs where it's a tube. However, once they exit the crypt and they're out on the villus, the shape now has to switch and the cells are convexly shaped. So they have an outward curvature. Now, we can take an even closer look at some of these cells that make up this intestinal epithelium. And the major cell type in the small intestine is the enterocyte. The enterocyte is covered in a dense brush border at its apical surface, and the brush border is made up of thousands of microvilli, which are very densely packed. This brush border is particularly important because it again allows for additional area for nutrient absorption, and it also provides yet another line of defense against pathogens, in addition to the mucus layer that we heard about earlier today. Now, if we take a closer look at an enterocyte where we're looking really closely at the ultrastructure within a cell, in this case, using a quick freeze deep HEM from the early 80s, what we see is that if we look directly underneath that cell membrane, at the apical surface, we have actin bundles, which form the core of microvilli, which then form the brush border. Those actin bundles plunge into this deep meshwork of filaments, which include intermediate filaments, which are these thicker filaments that will intermix with the actin bundles. And then there were these cross-linkers that were knew to be some type of myosin 2 fairly early on. In some of my earlier work in the TISCA lab, we discovered that non-muscle myosin 2C is very specifically enriched to the enterocyte terminal web, which I'm showing you here in these rather large scale images of these crypts and villi, where you can see that the non-muscle myosin 2C is really closely tied to specifically the apical surface of these enterocytes. It's not expressed in many other cell types in the intestine. And you can also see that there's a lot of F-actin structures that don't have 2C signal associated with it. These particular images are from a mouse that expresses an endogenously tagged GFP2C, which is a really great tool because non-muscle myosin 2C is expressed in relatively few tissues, just about 3 parts of the brain, the inner ear, and the intestine. And a lot of the antibodies targeted towards this protein are not particularly good. So we made a lot of use of this endogenously tagged mouse system. Now, I don't have much of an introduction to myosins in this presentation, but building off of what Zev and Thomas talked about, myosins are a contractile protein. They are very stereotypically associated with producing force and contractile force specifically. In the case of our enterocytes, this myosin 2 is localized to the base of these microvilli, where it appears to almost cross-link them, although I hesitate to use that wording. That's just what it appears from the fluorescent images. And we can see that there's even 2 populations of it. And we have a medial plate, which covers the entire apical surface of the enterocyte. It is filled with these dense puncta, and then we have a junctional population, which had been previously reported and is much brighter. However, some of our previous work demonstrated that non-muscle myosin 2C is responsible for some morphological changes at that ultrastructure level, where we're looking at individual microvilli and their lengths. What I want to talk a little bit about today is actually a non-muscle myosin 2C knockout animal, where we see really strange villar morphology. So now we're looking more at that physiological scale instead of the ultrastructure. So usually, if we're looking at villi within a mouse, we expect to see very stereotyped appearance. They're very finger-like, they're very similarly sized, although they do get shorter as you move from the proximal to the distal end of the intestine. But in general, you open up any mouse, you look in the gut, you know what those villi should look like, especially when you take a cross-section like this H&E Swiss roll. However, in our knockout animals, this was not the case. You lose that stereotyped appearance, and instead our villi take on a variety of shapes. They take on little fun protrusions, they seem to grow arms, they have these really deep buckled regions in the tissue. And in the case of this villus, it is in fact one villus on the very far right. It almost looks like 2 villi have fused together at the top, but they still have some space in between them at the bottom. All of these abnormal morphologies were very strange. They would appear across multiple animals at different ages. But something interesting happens when you take this into 3 dimensions. So this is a Swiss roll where we cut sections off a tissue block. These sections are only about 10 microns. So we really could be missing a lot of information by only looking at this in 2 dimensions. Instead, if we take this to 3 dimensions, and now we're looking at an entire chunk of tissue using some different imaging technologies that allow us to have a little bit more access to the full 3-dimensional scale of this tissue, we see that it's not just that these villi have arms or strange buckles in them, but really that the entire morphology of what makes something a villus has changed in a very dramatic fashion. Again, we've lost our stereotypical finger-like protrusions, which we can see in our wild-type villi, and now we've moved into things that really have a lot of different types of structures. This entire villus that I'm indicating to you with my cursor, that's actually all one villus. If you section through this Z stack, you see that that is indeed just one villus, despite the fact that it's massive. This one down here actually has a hole in the middle. It has a sort of atrium. And this one over here actually has 3 prongs. So if we think about what has been shown in the past, where we're thinking about this vilification of the gut, where we know that smooth muscle contractions take that initial embryonic gut tube and give it the villi that we think of, something has clearly gone awry in that process. But this protein that has been knocked out in these animals, non-muscle myosin 2C, is very specific to enterocytes, and it doesn't have any known role in any sort of developmental process. These animals are alive, they survive into adulthood, and they live very similar lifespans. But something else is happening with this tissue and how it's being organized that is completely changing the morphology. Not really something that the true extent of those morphological deformations were only something we could see in 3D. It wasn't simply just that something was protruding out, it was that the whole 3-dimensional structure of these villi have really changed in a dramatic fashion. This is just one example of how when you're looking at a structure like the intestine, 2 dimensions truly isn't sufficient to understand how these tissues are organized and truly how they function, because that 3-dimensionality is a key part of the function of the small intestine. So as we move into the future, 3-dimensionality is going to allow us a lot of opportunities for exploration. This is just an image that I took on our multi-photon microscope we currently have, and this is a view of what the intestinal crypts look like if we're looking at them through an intact gut tube. So these crypts are very complex structures. They don't just look like the simple tubes we might see in 2 dimensions. There's really a lot more dynamic processes going on here, and things just aren't as simple as they might seem if we're only looking at them in 2 dimensions. So as we move forward in the field, we really need to start thinking about, well, what are we missing if we take out a dimension? Whether that dimension is the Z dimension, whether it's time, what extra information can we gain about self-organization, about tissue migration, when we're truly looking at the organism as a whole? Of course, there's a few challenges that come along with this. One is that if we want to use mouse models, which we can do some in vivo imaging to look at cell migration, we need to have mouse models that are also genetically tagged with fluorescent proteins, which isn't necessarily an easy task to undertake. And it's also normally not a quick task to undertake. So we have to figure out some ways that we have access to genetic perturbations. Imaging technologies are also something that's going to need to adapt if we want to be looking at something with both time and with a great amount of Z depth. But a lot of these technologies are already available. They're just not completely built up to the extent that they might need to be. And lastly, one of the big ones is going to be analysis and data storage, particularly the data storage and handling, because once you start dealing with these files where you're looking at 350, 500, 1,000 microns worth of depth, and you're adding time-lapse information into that, you're getting files that are easily hundreds of gigabytes, and that's a very difficult thing to manage. Additionally, analysis of something where you're kind of at this mesoscale, where you're looking at maybe the shape of a crypt or the shape of a villus as opposed to the shape of a cell, there seems to be some gaps in how well our current citation technology is able to handle that. So that's really an area that in the future, I think, with more improvement, we'd just be able to get a lot more out of our data and be able to make more conclusions off of that. So with that, I'd like to thank Janelia again for this opportunity, and also thank my lab and all of the wonderful resources we have at Vanderbilt. And my dogs, because they know more about science than me at this point after a year of the pandemic.
Thank you so much, Colby. And I also want to thank our other two panelists, Thomas and Zev. We will now be going into the discussion and Q&A question period led by Patrick and David Klotman.
Great. Yes, those are three wonderful talks. I have to embarrassingly admit that whenever I hear sort of the term plasticity, I always think of the brain. But in fact, I think those three talks were great examples that epithelia is really far more plastic, and the dynamics are really, really important for sensing all of the sort of cues outside and then integrating that information to have some change. So with that, I think maybe we'll bring in a few audience questions here. If we could bring in G2 Mayor and Manu Prakash, I think they had two questions for Zev. And then there's also some audience questions. This is sort of a combination of two questions from Claudia Vasquez and Nick Cronin, which are similar. And Zev, they were kind of wondering about like where you see these distribution and changes between the mammary gland tissue. What do you think is driving those differences between the different mammary gland organizations that you see? Is there like cues coming from external cues, or is it an internal cue type of thing?
Yeah, I think that's a really fantastic question and sort of underlies what we're trying to do. It's a little bit difficult to get at that question in vivo because you can change composition and cell position by so many different mechanisms. And so one of the advantages of going to the model system, where we're working with real human cells but reconstituted in vitro, is that we can control for a lot of the parameters relating to cell-cell signaling and cell identity plasticity, and actually show that cell identity is fixed at the time scale of our experiment, and show that it's the underlying dynamics, the sort of activity in the system that's giving rise to the distribution. But as soon as you take that system and you move it in vivo, you now have to contend with the impacts of cell-cell signaling and the interplay between tissue structure and cell identity and state. And there, I think things start to get really, really interesting, and where really sort of new theoretical frameworks are necessary to understand what's going on. Because now those are linked, right? So cell state now becomes fundamentally a function of where it sits in the tissue, but where it sits in a tissue is itself fundamentally a function of cell state. So I hope that gets at your question.
Yeah, that's great. We can move on to Manu.
Yeah, Zev, I was just curious, and I think Jitu was asking the same thing. Have you tried exploring a broader range of, the challenge with equipartition is you have to use it very carefully. And what does it really mean, and teach us back about what might be happening in the system to begin with? So I'm just curious, if you start ramping up activity, or what does the activity landscape look like as another axis, where you start not just explaining something, but where this approach breaks down, because that's really what teaches us something new as well.
Yeah, so Manu, I wish I could give you a good answer to that question. What I can say is that in the sort of bottom-up system that we put together, where we build the cells into the tissue, we have quite explicit control over the interfacial mechanics by hitting specific molecules that we know regulate those. And we can also control this sort of the multiplicity of structural states by manipulating cell composition and tissue size. Activity is a much harder one, and we're trying to get at what's controlling that molecularly. And there's a large field of people studying, for example, these jamming, unjamming transitions in tissues, which I suspect is basically the same thing that we're studying here. And I can tell you that we find things that change activity. The vast majority of them also change mechanics, but there's some that do it in an interesting way. So, for example, we can show that inhibition of AKT has a big effect on activity in this tissue, and I couldn't tell you why. But it would be really wonderful, and I think this is a challenge for the field, which is that if it's cell behaviors or these sort of physical properties of a tissue that are contributing to tissue structure through self-organization, there's not necessarily a direct relationship between a specific molecule and the quantitative level of that behavior or property. It's sets of genes working together. So how do we identify how to manipulate those in a systematic way? I'd love your thoughts on that.
Yeah, so or anyone's thoughts if they'd like to step in, but I guess we can go with we've got 3 people with their hands raised, so I'll go from left to right here, which Michael Caplan, if you want to.
Thanks. Those were all great talks. So this question, I guess, is for Zev and Thomas. Zev, when I saw your talk, I was brought back to my college statistical mechanics and got the sense that what you were looking at looks very much like a partition function. But this, of course, is not a Markov process, right? It's going to be essentially a recursive process where each phase of stochastic event is going to create boundary conditions that are going to be deterministic or limiting for the next phase. And I guess what I'm wondering is what sort of dramatic potential energy well or attractor is created in these systems that allows this tremendous stochastic variation at the microscopic level to produce something that always looks more or less the same at a macroscopic level?
Yeah, this is a really good question. And it reminds me of a conversation I actually had with Maha a couple of years ago, where he very quickly dismissed my false notions of thinking about tissue self-organization as a sort of an energy landscape. So thank you for that, Maha. I guess what I would say is that in the system that we're looking at, we're trying to draw a box around a very specific step in self-organization. But in the context of development, exactly as you're saying, Michael, as you lay down a structural event that's coupled to changes in cell state, which are going to set a whole new stage in morphogenesis moving forward. So, here we're trying to be very specific about what we're looking at, and we can show that if cell state doesn't change dramatically over the course of the experiment, that the cells really are sort of sampling from a distribution of structures exactly as you would expect from a statistical mechanical system. It's obviously a completely different scale. It's very far from equilibrium, but it seems to be following those rules.
But if I can add to that, I wonder whether it's not the fact that you have different scales of organization. I mean, the fact that the cell groups actually create local environments at a larger scale actually does change the statistics of individual cells. So there's really interaction across scales probably is underlying this sort of, I don't know if you want to use canalization or sort of funneling of phenotypes towards something which is much lower dimensional than you would expect and maybe started with. So I wonder whether this sort of interaction across scales, whether in the context of shape or size control, is really underlying sort of the question you ask, Michael.
Yeah, I should also emphasize that we're focusing on the steady state. So the talk was not, the presentation was not addressing dynamics, which is another, or time evolution of the system outside the steady state.
David Clapham, do you want to, you've got your hand raised, do you want to?
Yeah, I'd like to ask a little more specific question. So I understand all the parameters you'd like to measure, and of course you cannot measure them all at all the scales necessary to understand how you build a building, let's say. But how useful has it been, or do you know anything about, for example, a simple process like cyst formation? Cysts form due to a lot of different stimuli, for example, due to immune stimulus or due to pathogen invasion, due to accumulation of fluid. So many different parameters, all of which don't seem to initiate the same kinds of cellular processes end with a cyst. Does anybody have any idea of why that is? Is that because the epithelia just have strong adhesions, stick together, keep growing side to side, and eventually form a circle? Or are there other steps involved? Does it initiate a program of some kind? And are such models like that may be useful to understanding how you change geometry and forces in epithelia? Anybody?
Like maybe we should pass that one to Maha, as he spent a lot of time studying cyst formation.
Or Michael, or
Yeah, so I don't know whether there's a general answer, but I think some of the things that you were raising do seem to be important. You know, the fact that if I have strong enough adhesion and if I have some sort of apical basal polarity, for example, which might give rise to curvature, then there is a potential to form a spheroid. And on top of that, if I now have some way to secrete, that's what's needed to essentially form cysts. How many different ways are there to get that polarity, and how many different ways are there to get secretion? I mean, I'm sure you know this much better than I do.
It seems like a common structural late adaptation, and it's not a simple. I mean, it's simple compared to, let's say, a breast, but it's not simple compared to, you know, other things. So it just puzzles me.
I think what you're raising
principles. Yep. Go ahead.
No, I think you're raising a different kind of question as well, which I was going to ask if I might, which is, in your systems, you know, all three speakers, you alluded to, at least some of you alluded to function. And I'm curious whether if you change the environment, which potentially is related or could be related to function, can you get different kinds of functional morphologies, either in the branching kind of situation, the cyst-like situation that we just heard about, or the folding-like situation. I mean, our own work over some period of time has shown that these same repetitive structures arise in very different settings, perhaps under very different kinds of controls, simply because the physics dictates that. You know, what can you do, kind of thing. But functions are very different. And so I'm just curious whether in your specific systems, can you tune the environment and or molecular, so either extreme of scale, stimuli to essentially guide structural rearrangements which are functionally relevant.
Yeah, I guess, yeah, I mean, well, that almost is a little bit, you know, one thing I brought away from Thomas's talk was just there's so much information that needs to be processed by the epithelia. And it seems like the largest challenge is how that information gets integrated to then make a change, and which information is the most important. And I don't know if anyone could speculate on, you know, how you might approach directly attacking one informational cue, or whether you approach attacking multiple at a time.
So if I could just jump in and follow up on David's question a little bit, and with apologies to Maha, and I recognize that viewing this as a potential energy landscape is a false metaphor, but it's possible that there are sort of metastable or primordial states that cells are especially comfortable in, and cyst may be one of them for some of the reasons that have been talked about, adhesion, simplicity of structure, et cetera. And getting to what David was alluding to, which is cysts as pathological processes. Cysts tend to be the common pathological outcome of a number of different disease processes, depending on which tissue you're looking in. And I'm wondering if what we're seeing is a sort of reversion to an earlier or stable state, metastable state of tissue biogenesis, where because cysts are essentially fairly stable or simple structures that are able to persist without a lot of other cues from the environment, that they can be common endpoints of a number of very different pathologies.
And that's sort of what I was getting to, Michael, is that it seems like it could be geometrically very stable, but also mechanically potentially very stable. But then how do you probe the system in all those different ways, inflammatory, et cetera, so that you end up with the same structure? Or maybe the cyst is an example where you don't need genes to make that structure, or some genetic network.
If I could say one thing about that, you know, the interesting thing about cysts that I think Maha was getting at is you really kind of only need two things for them to form. One is you need flux of liquid into a closed volume, Actually, it's really just one thing. You need a closed volume and flux of liquid into it. And when you think about some of the tissues that we've heard about today, whether it's the kidney or the intestine, in which you have regulated pumping of ions and liquids, typically a lot of those structures empty into an environment where the fluid can then flow or drain to another site, either be reabsorbed or leave the body. And a cyst is just, I feel like, is unique in that it's taking that fundamental aspect of epithelial biology, but it's just dumping the liquid sort of where it's not supposed to go. So that liquid can kind of build up.
Right.
But you also need a polarized distribution of basement membrane material and something on the apical pole that's different. I don't think you get cysts when the apical and basolateral components are the same.
Right. So if it could make an outlet, it would. If the geometry and forces were like an embryonic, you would think that it would form a canal out. Stimulate some program to do that. I don't want to dominate this whole thing with the cyst, but I thought it might be a useful example to say, what are the turning points for structural changes in these organs?
Interestingly enough, actually, the wing that I talked about is actually a cyst, but sort of flattened like a pancake. And it doesn't inflate like a balloon unless it's actually a mutant for things like integrins and other things. So it seems to be sort of an extreme variant of a cyst, one that actually is evolved to be just a flat sheet, but it's a cyst. An all imaginal disc, as a matter of fact, in insects.
So is the brain. So that's how the brain starts as well, right?
Yeah, that's how, exactly.
And so does the gut.
And maybe
let's see.
We are just a bag of cysts.
We're just spherical cows.
Maybe related to this conversation.
I think Elaine Fuchs has got a burning.
Yeah, I just wanted. Well, I have a question too for Zevin and Thomas together, but actually, just to follow up on this, it may be worthwhile considering that if you take an epidermal stem cell and you put it into a mammary fat pad, it will basically behave like a mammary epithelium, and it will form. It'll form a bilayered epithelium. If you take that mammary epithelial stem cell and you graft it into the skin, it will form a stratified epithelium. And so, just again, Thomas kind of finished on that note, but it's really you put it in the right environment, and I think there's a tremendous plasticity that is there, and it's something that you can change even postnatally. So worth to consider. But the question I had for Zevin and Thomas dealt with fluid, because as your fluid and proliferation, I mean, one, it's easy for you to just say, does your distribution change if you stop proliferation in the tissue with mitomycin C or whatever? But the other is what happens when fluid builds up in the lumen? Does that then impact on the overall distribution and tissue architecture and like? It would seem to me that it would.
Well, so there's an interesting example of the follicular epithelium in the fly ovary. So you have the oocyte is growing. So you indeed have, through endoreplication and dumping of the nerve cells into the oocyte, sort of huge increase in volume. And the follicular epithelium is a sort of somatic epithelium just around it. And actually, we had the talk before about the sort of integrin interactions with basement membrane of that tissue. But the point is that, so it's not been completely worked out by far, but there's some evidence that the growth, so the proliferation of the follicular epithelium is responding to actually the mechanical stimuli that emerge from the growth of the oocyte. There's been a couple of studies, but it's not well understood, but it sort of hints at the fact that there is indeed some mechanical connection between proliferation of the surrounding epithelium, this sort of ellipsoid, and the pressure that builds up from inside. And actually the work in the otic vesicles in the zebrafish that the Maha collaborated with Sean Magnuson on that also shows that there is a coupling. So usually people look at cell division, but the real issue is not cell division, but really cell growth. So I'm putting back into the equation sort of the questions of energetics there, which is at the heart of the problem. And I think it would be very interesting in those systems to look at whether, sort of the use of energy for growth is actually rerouted in the context where mechanics is applied to a tissue, where stress is applied or not.
And what about fluid? So that that addresses the issue of jamming or whatever, but then what about fluid buildup? breast milk or whatever. I mean, as you're producing like a lot, does that impact then on what the kinetics of the equation is going to look like with regards to the?
Well, I would tend to believe that, milk or other liquid is a bit like any cell. A cell is 70% water. So, the growth of an oocyte is essentially fluid that responds to protein synthesis. I'm not sure it has to be fundamentally different from the secretion of a liquid, which does produce a growth of an environment, although in that case it's probably 95% fluid indeed. Maybe at the mechanistic level there's no fundamental difference. Cells can respond and respond to the sort of the stress that emerge from the increase in volume.
So just to shift a little bit, Catherine and Helen had their hands raised for quite some time. So Catherine, would you like to start off?
Right, yeah, thank you. Beautiful talks. I first had a question for Colby and whether you've looked at the cycling of proteins along the microvilli and if that's perturbed in your knockouts as well. And then a broader question for Zev and Tomas. Sounds like you could use, or I guess a comment, model systems that have a really strong comparative strength to them, and I would recommend the echinoderms, so sea urchins and sea stars, because early in development, you can trace all of the cell contacts and the cell size over time, and different species have different associations of how tightly packed the cells are during development. And Vanessa Barone out of Dee Lyon's lab at SIO is doing beautiful work on that. She's not here today, but will be tomorrow. Okay, so back to the question, Colby. Do you see changes in transport of proteins along those villi? Thanks.
So do you mean transport of proteins along the microvilli, which is what you originally asked about? So
yes,
yeah, cargos. So broadly, no. We have some transcriptomic data that we're waiting on to see if there's something that we're missing just by imaging. But in general, other than these like big morphological changes and a few other changes that I'm not quite ready to talk about in public yet, these mice in general seem perfectly healthy. Several other groups have worked on them, and if you just were to take them at face value, you just had them in your mouse colony and you looked at them, you would think there's nothing wrong with them.
So have you tried challenging them in different ways? Because we see that in the transporter world for xenobiotic transporters, where the knockout the mouse looks fine, but then if you treat them with different drugs, they accumulate in the blood-brain barrier. Actually, collie dogs have a phenotype where they can't metabolize the tick medication, and so maybe you have to challenge them in different ways to see other phenotypes.
Yeah, we've considered challenging them, doing challenges with DSS or other very similar challenges that are very well known within the intestinal field. But just because in general, the focus of our lab is not very physiological. So these mice very well could also have some broader immune signaling that happens when we try to challenge them. But in general, we are much more interested in how this cytoskeletal motor that has a relatively small window of expression and a very specific part of the cell that it's localized to, how a change in that cytoskeletal molecule is influencing more of the morphology as opposed to that much broader immune signaling regimen and all of those other downstream effectors. It's just a little bit outside of the scope of what I'm currently working on.
Thank you.
Yeah, and I guess on that note, one thing that was particularly cool, Colby, was the just how specific the non-muscle myosin 2C is. So it really gives you like an opportunity to really assess, one protein and the function and morphology that follows, which I think is kind of rare. Great.
Yes, that is one of the big perks. Is it is extraordinarily specific, and within the intestine, it's extraordinarily specific even to enterocytes, which we do have RNA-seq data that backs that up. That it's not present in many of the other cell types in that tissue.
Helen, did you want to chime in?
Yeah, I mean, my question really is for, well, the feedback question. And with Colby, maybe you looked and maybe you didn't, but when you're seeing these really cool, I guess, non-fused, you're saying they're not fused, right? They're very malformed and
yeah, they're just, yeah, malformed is the best way to describe them.
Right. So what I'm curious about is, I don't even know what the time frame of this, but how does that feed back to sort of the development of villi? There's all these models about clustering of cells and, tensions. And so now you've got something that you might be perturbing the system. Have you looked back?
So we have, one of the things that I do want to do is I want to go back a little bit further. We generally do work with mice once they are 8 weeks old, once they have truly developed their crypts, their villi are very much mature. But there is obviously this big question of at what point are these perturbations happening? Is it something that's happening embryonically? Is it something that's happening neonatally? So I would like to go back and really figure out exactly the time point at which we see these perturbations. But at this point, we know that if the mice are developmentally mature at that 6-8 week mark, that we still see the same phenotype.
Right, so it's a stable phenotype, but I guess this gets back to our question of, sensing the forces. So here you're playing with the myosin up here, like what's happening everywhere else and mechanically, right, and as well, yeah.
Yes, exactly. And one thing I think that is important to note is that myosin is present across the entire surface of the enterocyte. It's also very present at the junctions, and we know junctions are mechanosensitive, and that on villi, those cells have to be able to move and migrate, and the junctions and the tension in the junctions play a very big role there. So I would make an educated guess that role of that tension, whether we now have excess tension because a different motor is in play, or we have less tension because we have less motors overall, that's playing a big role in why this tissue is getting stretched and deformed in the way that it is, because we can assume that not much else has changed. It's really just a fairly specific perturbation.
So how much else changed afterwards, right? That's the cool thing too.
Colby, along those lines, and just to bring it full circle to the first two talks of this morning, unless I missed what I was looking at, it looks like your knockout mice have many more goblet cells. So first of all, is that true? And second of all, how do you think that relates to the mechanical deformations that you were just talking about?
So yes, the mice do have quite a few more goblet cells. They also have a few more tough cells. And my best guess based on that, and just some other strangeness that is present in the tissue when we're working with it, is that there is a barrier defect because of that change in junctional tension. Just the barrier is not as effective as it needs to be, and that is causing a downstream inflammatory response. And that's why we see our goblet cell hyperplasia. That's why we see a certain extent of tough cell hyperplasia.
So I'm going to have to bring everything to a close. I'm sorry to have to stop this really exciting discussion, but we need to move to our panelist discussion. So with that, I want to thank all the panelists who've been involved in the first 4 sessions today. It's been super, super exciting. And I look forward to seeing everybody who's been listening to a full, exciting day tomorrow. For the panelists, however, we want you to stay on. We have a closed session, which is a separate Zoom link that you should go into now. And we will talk in more detail about some of the things that were discussed this morning. So thank you all, and we'll meet again tomorrow at 11 o'clock. Bye-bye.
Just to clarify, are all of us considered for this next session?
Yes, I'm gonna shoot an email out with the Zoom link.
Got it. Thank you.
Thank you.
Great, thanks.